When you hear the word prion, what is the very first image that flashes into your mind?
Oh, I'm guessing for most people it's an absolute medical nightmare.
Right, exactly.
Yeah.
If you are like almost everyone else on the planet, you are picturing something terrifying. You're thinking of a disease that is, untreatable, untraceable, and universally fatal,
Yeah, mad cow disease is the classic example that terrified everyone in the 90s. Or Creutzfeldt-Lehmann disease in humans.
which is just horrific to even read about.
It really is. I mean, the prevailing understanding for decades has been that prions are just these inescapable biological errors. It's a protein that misfolds, and somehow that purely mechanical misfolding becomes contagious,
Like it just cascades through the brain.
literally dismantling the neural architecture piece by piece. And the scary part is there's no virus, there's no bacteria. It's just a broken shape that forces everything around it to break in the exact same way.
It's basically the ultimate rogue agent in biology, a microscopic villain that we have spent billions of dollars trying to eradicate. But today, we are going completely through the looking glass on this deep dive. We are stepping into a world where that exact same villain, that indestructible, self-replicating, misfolded protein, is actually being hailed as the master blueprint for the next century of human infrastructure.
It's a massive paradigm shift.
It really is. Today's deep dive is focused on a sweeping, highly controversial, and frankly radical design manifesto by Zachary David Enfield.
Yeah, the title alone really tells you the scale of what we are dealing with. It's called Prion and Amyloid Architectures, a total application framework.
And when Enfield uses the word total, he means it in the most literal sense imaginable.
He really does. I mean, it is arguably one of the most comprehensive, paradigm-shattering syntheses of material science, quantum electronics, and synthetic biology that has been published in the last 20 years.
So our mission today is to take this incredibly dense framework, which is just packed with molecular physics and quantum theory, and break it down into something you can actually visualize.
Right, because the core premise is that these amyloid and prion structures are not merely medical anomalies to be feared.
Right, they are building blocks. We are going to explore how these exact microscopic biological fibers are positioned to literally replace the silicon in the computer or phone you are using right now.
From a purely physical standpoint, they represent the most information-dense, mechanically robust, and energy-efficient self-replicating nanostructures known to modern science.
Which is wild. And we're going to look at how they can function as atomic-scale nuclear batteries and how they could completely overwrite our entire civilizational infrastructure by the year 2060.
We are talking about fundamentally abandoning the idea of building our technology and transitioning entirely to growing it.
Which is just a mind-bending concept for you to think about. But before we even begin to comprehend how a protein could replace a silicon computer chip, we have to look closely at the structural reality of what an amyloid prion actually is.
Yeah, because the way this thing is constructed at the atomic level dictates absolutely everything else it is capable of doing. Biology is destiny here, but, in a highly engineered sense.
Let's start right at the bedrock.
Huh.
Enfield bases this entire technological revolution on something he calls the cross-beta architecture.
Right.
If we had a microscope powerful enough to zoom all the way down to the 10-nanometer scale, what are we physically looking at?
Well, you would see a highly specific, almost crystalline repeating pattern. The cross-beta architecture is a structural motif where you have individual protein chains.
Okay.
And these are referred to as beta strands. Imagine them as short, relatively straight ribbons of amino acids.
Got it. So little ribbons.
Yeah, and in an amyloid fiber, these individual ribbons stack up perfectly on top of one another, like the rungs of a very long ladder.
Okay, that makes sense.
And crucially, these rungs are oriented perpendicular to the long axis of the growing fiber.
So if the fiber is growing straight up toward the ceiling, the beta strands are stacked horizontally, flat against each other, building the tower upward.
Correct. But the real magic lies in how those flat strands are glued together.
What's holding them?
Running vertically parallel to that long axis is an incredibly dense, continuous network of hydrogen bonds.
Oh, wow.
Yeah, the hydrogen bonds connect the backbone of one beta strand directly to the backbone of the strand immediately above it and below it.
It makes me think of a self-assembling zipper or like a microscopic Lego tower where every single piece snaps into the exact same orientation with a very satisfying click.
That's a great way to picture it.
But it can't just be a rigid, brittle tower, right? The manifesto spends a lot of time discussing how these structures have alternating layers.
Yes,
You
exactly.
have the rigid, hydrogen-bonded beta sheets in the core, but they are separated by what Enfield calls side chains that act like physical shock absorbers.
And that is the nanocomposite genius of the structure. When you stack those beta strands, the side chains of the amino acids interlock.
Like the parts of the molecule that stick out from the main backbone.
Right. They interlock like the teeth of a gear. So you have this immense, unyielding stiffness provided by the vertical hydrogen bonds, but it is immediately coupled with the energy dissipating, flexible nature of those interlocking side chains.
So it's strong, but it can flex.
Exactly. It is the exact same mechanical logic that nature uses to build nacre.
Nacre.
Yeah, the incredibly tough, iridescent mother of pearl inside an abalone shell, or even the high tensile tendons in your own body.
Oh, I see.
But here, that logic is operating cleanly at the absolute molecular scale.
Which brings us to a massively important concept in the framework. It's a metric called hydrogen bond density, or HBD.
Yes, HBD is crucial.
From reading the manifesto, HBD seems to be the ultimate design dial for the engineers building these things.
It is. HBD is the master control knob for the entire physical universe of amyloid materials. The mechanical properties of these fibrils, their tensile strength, their elasticity, their shear resistance, are directly proportional to it.
Proportional to the density of the hydrogen bonds packed into the core.
Exactly. Mathematically proportional. If an engineer wants to create an incredibly rigid structural material, something intended to replace structural steel beams in a building or the chassis of a vehicle, they look at the genetic sequence.
And they just tweak the protein.
Yeah, they tweak it to pack those residues as tightly together as physical chemistry allows. They maximize the HBD.
And conversely, if they want something highly dynamic,
They dial the HBD down. By designing a protein sequence that incorporates disordered or highly polar regions in the core, the hydrogen bonding network becomes looser.
so it gets weaker.
The resulting fibril becomes fragile, yes. It breaks easily under mechanical stress, which counterintuitively is exactly what you want if your goal is rapid, explosive self-replication.
Right, because breaking creates more fiber ends to grow from.
Yeah.
We will get into that replication cascade in a second, but I want to linger on the sheer physical strength of the high HBD materials for a moment.
The numbers are pretty wild.
Because the numbers Enfield cites are just staggering. He points to data on engineered polymeric amyloid fibers, specifically a sequence known as FGAILSS.
Ah, yes.
And I had to look this up. Those letters are just the single-letter codes for the specific amino acids in the chain, right?
Yep. Phenylalanine, glycine, alanine, isoleucine, leucine, serine, serine.
Right. So when they synthesize that specific sequence and let it assemble, the resulting material has a toughness of 160 megajoules per cubic meter.
That number requires context to truly appreciate.
Exactly. For context, natural spider dragline silk, which is universally considered the holy grail, the absolute gold standard for biological toughness, maxes out lower than that.
Way lower.
This engineered protein fiber beats spider silk. And it does this while weighing only 1/8 as much as high tensile steel.
It is a profound, almost unbelievable combination of physical properties. You are looking at a material that is measurably tougher than spider silk, a fraction of the weight of industrial steel, and entirely biocompatible.
Because it's just made of amino acids.
Right. And completely infinitely tunable just by rearranging the letters of its sequence.
You're painting a picture of a miracle material here. But as a reader, my brain immediately rebels against this.
What else so?
Because when I think of a biological protein, I think of a piece of meat left out on a counter. I think of something that rots. It melts.
Sure.
It degrades under UV light. It is consumed by bacteria. If we are genuinely talking about replacing global industrial infrastructure, the steel in our bridges, the protective casings of our electronics, with protein fibers, how does this stuff not just turn into a puddle of mush the first time it rains?
Right, because biology is inherently fragile outside of a living organism.
Exactly.
That is the intuitive, everyday assumption we all make, and Enfield systematically dismantles it. He introduces what he calls the indestructibility factor.
Okay, color me skeptical.
Well, it turns out that the specific cross-beta structure we are talking about survives conditions that would instantly and violently obliterate normal, functional biology.
What kind of conditions?
The source cites extreme laboratory stress tests. Fibrils formed from these high HBD sequences survive being immersed and boiled in concentrated sodium hydroxide.
Wait, boiling acid.
They remain completely structurally intact after prolonged exposure to pure boiling formic acid.
You drop a biological protein into boiling acid, and it just sits there.
Hmm.
How does the acid not dissolve the chemical bonds?
Because of steric exclusion and the thermodynamic minimum of the fold?
Translate that for me.
Basically, the cross-beta structure is so densely packed and the hydrogen bonds are woven so tightly that the physical structure of the fiber actually shields the vulnerable peptide bonds.
Oh, it acts like an armor.
Yes. Water molecules and corrosive acid ions physically cannot penetrate the core of the fiber to break the chain. Furthermore, they are entirely resistant to protease enzymes.
Proteases are the biological scissors, right? The enzymes in our stomachs that digest the proteins we eat.
Exactly. Normally, a protease lashes onto a protein strand, finds a specific sequence, and snips it in half.
But they can't cut this.
The protease enzymes literally cannot find a purchase on the smooth, tightly locked exterior of the amyloid fiber. It is like trying to cut a solid diamond with a pair of plastic safety scissors.
That is insane.
But it goes far beyond chemical and biological resistance. These structures are radically radiation hardened.
Which is vital if they're going to replace electronics.
Precisely. Experimental data shows that prion proteins can withstand doses of gamma-ray radiation that would instantly fry the crystalline lattice of a silicon logic board.
Or completely embrittle copper conductors, right?
Yeah. Silicon relies on a delicate, perfectly ordered atomic lattice to conduct electrons. High-energy gamma radiation knocks silicon atoms completely out of position, creating defects that destroy the chip.
But the protein doesn't care.
Because the amyloid fiber is a soft matter nanocomposite. When radiation strikes it, the energy is dispersed through the flexible side chains as vibrational heat. The hydrogen bonds might momentarily stretch, but they immediately snap back into place.
So we have a physical material that we can dial in to be tougher than spider silk, lighter than steel, impervious to boiling acid, and capable of shaking off gamma rays.
That's the summary, yeah.
But you mentioned earlier that the most disruptive property isn't its strength. It's the manufacturing paradigm. It is self-replicating.
This is the real game changer.
And this is where my mind really bends. Because a protein is a dead chemical. It is not a cell. It doesn't have a nucleus or its own DNA to divide and conquer. How does a dead chemical string self-replicate?
It replicates through a purely thermodynamic process called a seeding cascade.
Okay, walk me through that.
To visualize this, imagine you have a massive industrial vat of liquid. Inside this liquid, there are trillions of loose, unassembled monomer proteins.
Just floating around.
They're just floating around randomly. In this state, they are completely harmless and structurally useless. They don't form fibers on their own because there is a high kinetic energy barrier preventing them from falling into that specific cross-beta fold.
They need a push.
They need a template. The moment you introduce one microscopic fragment of an already correctly folded amyloid fiber into that vat, the seed, everything changes.
So you just drop the seed in.
Yeah, the seed acts as a physical mold. The loose, random proteins bump into the exposed end of that seed via normal Brownian motion.
Just randomly knocking into it.
But when they touch the exposed interface, the specific geometric arrangement of the seed exerts an electrostatic and steric force on the loose protein, forcing it to instantly snap into the exact same cross-beta shape.
It's like a chain reaction of origami.
That's exactly it.
You have a room full of flat, scattered pieces of paper. One perfectly folded origami crane touches a flat piece of paper, and the physical contact alone forces the flat paper to fold itself into an identical crane, completely automatically.
That is a brilliant way to conceptualize template-directed templating. The newly folded protein is now part of the fiber, extending its length and creating a new exposed surface for the next random protein to bump into.
And it just keeps going.
The fiber grows perfectly straight, molecule by molecule.
But if it just grows straight, you just end up with one incredibly long microscopic string. How does the population actually multiply?
Through the mechanical fragility we discussed earlier.
Oh, the low HBD dial.
Right. As the fiber grows longer and longer, the ambient mechanical agitation in the liquid, just the currents of the liquid being stirred, causes the long fiber to snap.
It breaks.
It fragments. Now, instead of one growing end, you have two. Those two grow and then snap, creating 4, then 8, then 16.
Oh, wow. So it's exponential.
The population of growing fiber ends increases exponentially. It converts the entire vat of raw protein feedstock into identical, indestructible fibers.
And the most mind-blowing part is the energy equation here. In a steel mill, we burn tons of coal to melt iron and poured it into molds. Here, the energy input from the outside world is almost zero.
You literally just stir the vat.
The thermodynamic energy released by the proteins falling into their most stable shape drives the entire manufacturing process.
Material manufactures itself. That is the foundational premise of Enfield's manifesto.
Okay, so we have established the physical reality of these structures. We have this indestructible, self-replicating, self-assembling fiber.
Yes.
But how do we get from a very tough piece of biological string to replacing the computer processor that you, the listener, are using right now? That leap feels incredibly vast.
It does, until we examine the physical bottleneck of modern computing. We are reaching the absolute physical limits of what silicon can do.
Right. Moore's law is dying.
It is dying because of atomic reality. A silicon chip operates by pushing electrons through physical pathways and blocking them with physical gates to create ones and zeros.
Just standard binary.
Exactly. Over the last 50 years, we have made those pathways and gates smaller and smaller to pack more computing power onto a single chip. But we are now at a scale where the gates are only a few dozen atoms wide.
Which is unimaginably tiny.
And at the sub-nanometer scale, quantum mechanics takes over. Electrons simply start jumping straight through the closed gates via quantum tunneling?
The silicon can no longer contain the charge.
Right. And even when it does contain the charge, pushing all of that current through microscopic atomic bottlenecks generates immense friction.
Unmanageable amounts of waste heat. This is why major tech companies are building data centers next to rivers or in the Arctic.
The energy required to cool the silicon is fast becoming greater than the energy required to run the computations. Silicon has hit a thermal and quantum wall.
And Enfield's framework declares that these self-replicating prion fibers are the solution to that wall.
They bypass it entirely.
But my immediate roadblock here is conductivity. You have a protein fiber. Protein, by definition, is an organic insulator. It does not conduct electricity.
Traditionally, no.
So to make a computer out of this, you would have to use the protein as a scaffold and coat it in copper or gold.
You certainly can coat it in metal. And historically, that was the first step. Enfield notes a landmark study from way back in 2003, published in PNAS, where researchers used a yeast prion called SUP35 to grow a self-assembled fiber. They then used that fiber as a biological template to chemically deposit gold nanoparticles, creating a perfectly continuous, highly conductive nanoscale wire.
Oh, that's clever.
The genius of that 2003 paper was that they grew a nanowire in a beaker at room temperature, completely bypassing the need for multi-billion-dollar clean rooms and toxic photolithography chemicals.
That is amazing in itself, just as a manufacturing shortcut.
Yeah.
But the manifesto goes so much further than that. You don't actually have to coat these biological fibers in metal at all.
Not anymore, no.
The source references a much more recent breakthrough, a 2021 study by Ships et al. That demonstrated these purely biological fibers can conduct electricity intrinsically, all by themselves.
That's a massive leap.
How is that physically possible? There are no free electrons floating around in a protein like there are in a copper wire.
It relies on a quantum mechanical process known as tyrosine hole hopping.
Tyrosine hole hopping, break that down.
Tyrosine is a specific amino acid. Its molecular structure features a large hexagonal aromatic ring.
Okay, a hexagon.
When engineers design an amyloid sequence with a high concentration of tyrosine residues, something remarkable happens during the seeding cascade. As the cross-beta architecture folds together, those hexagonal tyrosine rings stack up perfectly, one on top of the other, vertically along the central core of the fiber.
Like a stack of coins running down the middle of a tube.
Exactly. And the distance between these stacked rings is incredibly small.
Yeah.
Roughly 3.5 angstroms.
Which is basically Dutching.
At that proximity, the electron clouds of the aromatic rings actually overlap. They merge into a continuous, delocalized pi-stacked pathway.
Okay, let me visualize this. You have this stack of hexagonal rings, and their electron clouds are blurring together into one continuous molecular tunnel.
Yes, and when you apply an electrical bias to one end of the fiber, an electron isn't flowing smoothly like water through a pipe, which is how ballistic transport works in a copper wire.
So how does it move?
Instead, the charge hops from one ring to the next via quantum tunneling. The overlap of the pi electron clouds drastically lowers the energy barrier for the hop.
Wow.
It allows electrical charge to zip down the length of the biological fiber over micrometer distances with startling efficiency.
It is like passing buckets of water down a fire line, but the firefighters are spaced perfectly at the quantum level, so the buckets just teleport from hand to hand.
That's exactly how hole hopping functions.
So the pure protein fiber itself is the wire.
It is a self-growing, indestructible nanowire.
But a wire is just a conductor. A wire is not a computer. To actually process information, you need memory to store data, and you need logic gates to manipulate that data.
You need the whole architecture.
And this brings us to what I think is the most purely science fiction concept in the entire manifesto. Enfield proposes something he calls the computational soup. He is outlining a hybrid DNA prion processor.
It is a radical departure from the von Neumann architecture we have used for 70 years.
Uh,
Let's break down the components of this hybrid. First, you need mass storage.
Right.
In a traditional silicon computer, storage is strictly binary. It uses electrical voltages to represent zeros and ones. In Enfield's biological computer, the storage medium is DNA.
Which makes sense because DNA is nature's ultimate hard drive.
More than that, DNA inherently utilizes quaternary logic. Instead of 2 states, it has 4 fundamental states corresponding to its nucleobases.
Adenine, cytosine, thymine, and guanine A, C, T, and G.
Exactly.
So what does that actually mean for the math of computing?
It means that every single physical state change in a DNA molecule stores 2 bits of information, 0, 0, 0, 0, 1, 10, or 11, instead of the single bit stored by a silicon transistor.
You are doubling your data throughput at the exact same clock speed purely through the base 4 mathematical architecture.
Yes. Furthermore, a silicon wafer is a 2-dimensional flat surface. You can only etch data onto the top layer. DNA is a 3-dimensional molecule suspended in a volume.
The density must be insane.
The information density per cubic millimeter of a 3D DNA matrix completely obliterates anything you could ever theoretically build out of 2D silicon wafers.
We're talking about storing the entire data footprint of the modern internet in a volume the size of a shoebox.
Easily.
But here's the historical catch. DNA computing is not a new idea. We've known DNA can store data for decades. The problem has always been the clock speed.
Reading and writing data.
Right. Reading and writing data to DNA usually requires chemical reactions, synthesizing strands, or using enzymes to cut and paste. And chemical reactions are agonizingly slow. It takes hours to process what a silicon chip does in a microsecond. How does the computational soup solve the speed problem?
This is where Enfield integrates the third discipline, quantum photonics. The manifesto discards chemical enzymes entirely.
No more enzymes.
To achieve modern clock speeds, the framework utilizes intense pulses of terahertz radiation.
Terahertz. We're talking about the frequency band sandwiched right between microwaves and infrared light on the electromagnetic spectrum.
Yes. When you direct highly calibrated, ultra-fast pulses of terahertz radiation at a DNA double helix, the photons resonantly excite the collective vibrational modes of the hydrogen bonds holding the AT and CG pairs together.
You vibrate the bonds until they let go.
Exactly. The terahertz pulse physically unzips the specific section of DNA in a matter of picoseconds, trillionths of a second.
That's incredibly fast.
It separates the strands, exposing the bases to be read or modified, and then a subsequent pulse, or simply the removal of the field, allows it to rapidly rezip. You are using light to toggle the molecular state at terahertz clock speeds?
Okay, so the DNA is your high-speed, incredibly dense quaternary hard drive. But you still have to wire those individual DNA memory sites together to perform logic operations.
Right, the interconnects.
You can't take a macroscopic copper wire and solder it to a nanometer-scale DNA molecule. The scale mismatch is like trying to connect a fire hose to a hypodermic needle.
And that is exactly where our indestructible prion fibers re-enter the narrative. The amyloid fibers act as the physical interconnects, the wiring between the DNA nodes.
They hook it all up.
Because the fibers are roughly 10 nanometers wide, they are perfectly scaled to interface natively with the DNA. Through specific molecular binding domains, the prion fibers self-assemble.
They grow toward it.
They literally grow outward from one DNA node, navigate the liquid medium, and bind to an adjacent DNA node. The intersections of these fibers create the physical logic gates, transferring the whole hopping electrical state from one memory locus to another.
I just love the visual analogy for this. Current silicon computing is like a rigid, massive 2D highway system made of concrete. You have to lay down the roads perfectly in advance inside a multi-billion-dollar sterile factory.
It's very rigid.
But this DNA prion processor is fundamentally different. It is like a self-growing, three-dimensional organic brain floating in a jar of liquid. It wires itself together dynamically.
It is a unified molecular computation medium operating entirely at the sub-nanometer scale. But, your analogy highlights the exact physical hurdle that skeptics of this theory always point to.
The jar, the liquid environment.
Exactly.
Yes. When I read about the terahertz unzipping mechanism, red flags went off everywhere. If you know anything about the physics of terahertz radiation, you know it has a fatal, well-documented flaw.
Water.
Liquid water absorbs terahertz waves like a black hole. It is highly opaque to those frequencies. If this entire computational soup is floating in a biological liquid bath, the water molecules are going to absorb 100% of the terahertz radiation before it ever travels a single millimeter.
The signal would die instantly.
The unzipping signal will just vanish. How does Enfield's manifesto address the water problem?
Enfield acknowledges that blasting the entire bulk liquid with a raw terahertz beam is thermodynamically impossible. The proposed solution is a highly advanced technique called nanoplasmonic focusing.
Unpack that for us.
The architecture does not rely on bathing the entire system in radiation. Instead, the framework proposes embedding tiny, nanoscale antennas directly into the biological matrix.
Antennas inside the soup.
These antennas are made of specific plasmonic materials, bismuth or gold nanoparticles.
But wait, I have to interrupt there. How do you get precisely shaped gold antennas perfectly embedded into a self-growing biological soup without ruining the grow-it-in-a-vat manufacturing paradigm?
Through the exact same self-assembly logic. The biological sequence of the amyloid fiber is engineered to contain metal-binding peptide sequences.
Oh, come on.
As the fiber grows, it scavenges trace gold ions from the solution and physically organizes them into geometric nanoantenna clusters at specific nodes along the fiber.
The biological fiber builds its own metal antennas as it grows.
Precisely. And when the external terahertz pulse is fired into the medium, these embedded bismuth or gold antennas capture the diffuse terahertz field.
And they funnel it.
Through surface plasmon resonance, they compress and focus that electromagnetic energy down to an intense sub-wavelength pinpoint, directing it exclusively onto the dry, hydrophobic core of the specific DNA strand they are attached to.
That is cheating physics in the best way possible.
You completely bypass the water absorption problem by localizing the energy exactly, and only where the unzipping needs to happen.
Wow. Okay, so you cheat the water absorption by turning the fibers into fiber optic lightning rods. But that leads to the next massive objection: heat.
Thermal limits.
We established that silicon chips fry themselves with waste heat. If we are doing trillions of DNA unzipping operations every second using terahertz radiation inside a dense biological soup, how does the water not literally boil?
This brings us to what I consider the most elegant and arguably the most important pillar of Enfield's architecture: thermodynamic computing.
Thermodynamic computing.
This represents a total inversion of how we think about energy and technology.
How so?
Silicon engineers view heat as the ultimate enemy. They spend fortunes trying to pump heat out of processors. But the manifesto integrates findings from a seminal 2025 paper published by researchers at Caltech in the journal Nature.
Okay.
They demonstrated that in a liquid-state molecular computer, you can actually use ambient heat cycles to recharge the logic circuit.
Recharging a computer with heat. The heat is the battery.
Essentially, yes. And to understand why, we have to look at the physics of information erasure.
Erasing data.
In a conventional silicon logic gate, an AND gate or an OR gate, two inputs come in and one output leaves. To do that, the gate must physically erase the memory of the inputs. In 1961, physicist Rolf Landauer proved that the physical erasure of one bit of information intrinsically and unavoidably releases a specific tiny amount of heat energy into the universe.
It is a fundamental law of thermodynamics.
Yeah.
Erasing data makes heat.
Because you are decreasing the entropy of the computing system, so the entropy of the surrounding environment has to increase as heat.
Right.
But the DNA prion architecture is designed as a reversible computing system. It does not erase inputs, it simply changes its physical geometry.
Wait, how does that work?
When the terahertz pulse hits the nanoantenna, it causes a physical conformational change in the DNA molecule and unzips. That spatial change alters the electrical resistance of the attached prion fiber, which represents the new logic state.
It just changes shape.
The information is not destroyed, it is conserved in the newly adopted geometry of the molecular complex.
It is like manipulating an abacus. Sliding a wooden bead from left to right doesn't destroy the bead, it just changes its physical location to represent a new number.
That's a great way to think about it.
And moving a bead creates virtually zero waste heat compared to burning a piece of paper with a number written on it.
That is a perfect analogy. The system operates remarkably close to the theoretical Landauer limit of 100% thermodynamic efficiency.
Because it's just moving things around.
Because the computation relies on geometry rather than erasure, the ambient thermal energy of the liquid bath, the heat generated by the slight friction of the molecules, actually provides the vital equilibration energy needed to stabilize the molecule in its new calculated shape.
The thermal environment isn't the enemy. It is the power source driving the next calculation.
Which means the energy consumption of a global data infrastructure built on this architecture would drop by multiple orders of magnitude.
Okay, my mind is thoroughly expanded on the physics. We have a hyper-efficient, liquid-state, heat-powered biological computer.
We do.
But that raises a massive logistical nightmare. We established in Section 1 that this stuff replicates exponentially through a templated seeding cascade. If you have a vat of raw protein monomers and you drop a seed in, it replicates uncontrollably in all directions.
It's unchecked exponential growth.
How on earth do you program the physical shape of the computer? If you just let it grow, you don't get a beautifully ordered microprocessor, you get a microscopic hairball. How do engineers actually dictate where the wires go and where the logic gates form?
You don't build the architecture physically, you write the matter into existence using light.
Writing matter with light. Okay, Enfield refers to this as prion optogenetic lithography.
Yes.
Let's meticulously unpack this. I am familiar with optogenetics from neuroscience, which involves making proteins sensitive to light.
Yes, the foundational toolset is borrowed directly from neurobiology. Nature has evolved dozens of light-responsive protein domains molecules like Cry2 or LOV2.
Where do those come from?
They are originally found in plants and algae to help them track sunlight. When a photon of a specific wavelength hits these proteins, they undergo an instant reversible change in their physical shape.
So how does Enfield apply a plant's sunlight-tracking protein to a self-replicating prion?
Through genetic fusion, engineers take the genetic code for the incredibly tough, self-assembling amyloid sequence, and they physically fuse it to the genetic code of the LOV2 optogenetic domain.
They staple them together.
They create a chimeric two-part molecule, and here is the genius of the design. In the dark, the LOV2 domain is tightly folded up. It acts as a physical, steric straitjacket.
A straitjacket.
It wraps around the prion sequence and completely prevents it from falling into the cross-beta architecture.
So you can have a massive million-gallon industrial vat completely full of these chimeric monomers. You can stir it, you can heat it, you can agitate it, but as long as the vat is kept in the dark, nothing happens.
They are locked. The seeding cascade cannot begin.
They are biologically inert.
But when you expose that specific volume of liquid to a highly calibrated wavelength of light, say a 470 nanometer blue laser, the photochemistry engages.
The light hits it.
The LOV2 domain absorbs the blue photon. The energy breaks a specific cysteine-flavin chemical bond inside the domain, causing an alpha helix to forcibly unwind. The straitjacket violently pops open,
Releasing the prion domain.
the prion domain is suddenly free to adopt its cross-beta fold. The seeding cascade instantly triggers.
Oh, wow.
But, and this is the crux of the entire manufacturing paradigm, the cascade only propagates in the exact submicroscopic volume of liquid where the laser light physically intersected with the molecules.
This is unbelievable. It is functionally a 3D printer, but there is no print head.
No mechanical parts at all.
There is no nozzle extruding hot plastic layer by slow layer. Instead, you have a completely still pool of liquid protein, and you shine a highly complex holographic laser projection into the depth of the pool, and the liquid literally freezes into solid, indestructible, conductive architecture instantly.
Exactly, and only where the light touched.
You are sculpting matter with photons.
And the physical resolution of that sculpt is limited strictly by the diffraction limit of the light you employ.
So it can be incredibly detailed.
If engineers utilize extreme ultraviolet light, combined with two photon polymerization techniques where two intersecting lasers are required to unlock a single molecule, they can pattern these massive three-dimensional structures with resolutions down to tens of nanometers.
That's chip scale.
They can sweep a laser matrix through the growth medium and literally draw the DNA logic gates, draw the conductive amyloid interconnects, and draw the structural support beams, all in one continuous fluid operation.
It is the ultimate realization of software dictating hardware. So what are the immediate real-world applications of this optogenetic lithography? Because the manifesto highlights an area that caught me completely off guard. Food tech.
It is positioned as the final disruption of global animal agriculture.
Tell me more.
The fatal flaw of the current plant-based meat industry is texture. They can synthesize the flavor profiles, but they cannot replicate the complex microarchitecture of real bovine muscle tissue.
Right.
Muscle is not a paste. It is an incredibly intricate, hierarchically aligned matrix of protein fibers and connective tissues.
A burger is just ground up, but a steak has grain.
Mhm.
It has a physical structure that our teeth understand.
Exactly. With prion optogenetic lithography, engineers can execute what Enfield calls protein lattice printing.
Okay.
You utilize safe, specifically designed, completely non-pathogenic amyloid sequences that digest perfectly in the human stomach. You fill a bioreactor with this liquid feedstock.
Mhm.
Then, you shine a holographic light pattern into the reactor that is an exact nanometer-perfect replica of the microarchitecture of a Wagyu steak.
You literally grow the steak, voxel by 3D voxel, using light.
The light triggers the cascade, and the liquid protein instantly self-assembles into the exact geometric aligned lattice of real muscle tissue.
That's just wild to think about.
It provides the authentic chew, the resistance, and the snap of real meat, built flawlessly from the molecular level up. You achieve this without raising a single animal, without utilizing a single acre of grazing land, and with a manufacturing energy cost that approaches zero.
That alone is a multi-trillion-dollar civilizational shift. But the framework, incredibly, goes much further and into much deeper ethical and biological waters.
It does.
This brings us to the application Enfield calls the synthetic cortical processor. He proposes using this light-writing technology to grow a brain-computer interface directly inside the living tissue of a human brain.
It represents the ultimate therapeutic and augmentative application of the technology. To understand why it's necessary, look at conventional brain-computer interfaces.
The neural laces and chips being physically implanted in patients today.
Right. They involve drilling a hole in the skull and physically driving rigid silicon or metal electrodes deep into the unimaginably soft, gelatinous tissue of the cerebral cortex.
Which is incredibly traumatic. The brain's immune system immediately recognizes the rigid silicon as a foreign invader.
It fights back.
It attacks the electrode, encapsulates it in thick glial scar tissue, and over the course of a few years, the scar tissue completely blocks the electrical signal. The implant goes deaf.
It is a fundamental mechanical mismatch between rigid machinery and soft biology. But when I read Enfield's solution injecting a synthetic, self-assembling, indestructible protein cascade directly into a patient's brain, I shared your exact reaction.
It sounds like intentionally giving someone a superpowered version of Alzheimer's. How does the brain not immediately launch a massive neuroimmune response to reject this growing web of fibers? How is a synthetic protein mesh any better than a silicon electrode?
Because of a biological reality that Enfield calls the native brain bridge. It is a little discussed fact outside of neuroscience, but the healthy human brain already relies heavily on cross-beta amyloid structures to function.
Wait, really? We have amyloids in our brains right now. I thought amyloids were exclusively the plaques that cause dementia.
They are implicated in neurodegenerative diseases when they misfold pathologically and form massive, suffocating plaques. But in their normal, physiological, correctly managed state, they are vital.
Vital how?
Proteins like tau, which stabilizes the long microtubule highways inside your axons, and alpha-synuclein, which helps regulate neurotransmitter release, both natively utilize ordered fibrillar structures. They are part of the brain's existing healthy structural matrix.
That changes the paradigm completely. So how does the synthetic cortical processor interface with that existing healthy matrix?
You do not physically inject a pre-assembled fiber. You utilize standard gene therapy techniques.
Okay, so viral vectors.
You take an adeno-associated virus, an AAV vector, which has been stripped of its viral payload and loaded with the genetic code for our engineered, optogenetically locked prion protein.
You inject the blueprint.
You deliver this vector to the specific target region of the cortex. The virus harmlessly enters the patient's neurons and drops off the blueprint.
So the neuron itself becomes the factory.
Yes. The neuron uses its own ribosomes to manufacture the synthetic protein. The newly minted proteins sit quietly in the cytoplasm, entirely harmless.
Because they're locked.
Locked in their straitjackets by the LOV2 domain because it is completely dark inside the skull.
Right.
Then the physician initiates the assembly. They shine a highly specific pattern of near-infrared light, which can penetrate through the skull and deep into brain tissue, defining the exact neural circuit they wish to augment.
The infrared light hits the target neurons, the optogenetic straitjackets pop open, and the seeding cascade begins.
But crucially, they do not assemble into an isolated rogue clump. The synthetic sequence has been exquisitely engineered to recognize and chemically co-assemble with the native tau and alpha-synuclein scaffolds already clinging to the neuron's cytoskeleton.
It piggybacks on them.
The synthetic computing matrix literally weaves itself seamlessly into the existing physical architecture of the brain. It is expressed from the neuron's own genome, built by the neuron's own machinery, and anchored to its native scaffolding.
That's why there's no immune response.
The brain's immune system, the microglia, does not attack it or form scar tissue because, molecularly speaking, it belongs there.
That is fiercely elegant. It is terrifying to contemplate, but from a purely engineering standpoint, it is a masterpiece. But here's the lingering mechanical question.
Sure.
If you don't have rigid silicon electrodes physically touching the synapses, and you don't have a coin battery implanted under the scalp, how does this synthetic mesh actually read the data? How does it pull your thoughts or motor commands out of the brain to control a computer or a prosthetic arm?
It reads the data through a quantum phenomenon called electron spin resonance, or ESR.
Electron spin resonance.
Every time a neuron in your brain fires an action potential, that electrical spike generates a vanishingly tiny, localized, time-varying magnetic field.
Because moving charges create magnetic fields. The brain is electromagnetically noisy.
Exactly. Now, recall the intrinsic conductivity of our synthetic prion fibers from Section 2? We discussed how the aromatic rings of the tyrosine and tryptophan residues form a continuous overlapping electron cloud down the core of the fiber.
The pi-stacked pathway for the hole hopping.
Yes, the electron spin states within that highly ordered pi-stacked cloud are hypersensitive to external magnetic fields.
Ah, I see where this is going.
As the native neuron fires and generates its tiny magnetic pulse, that field washes over the intertwined synthetic fiber. The magnetic field instantly alters the electron spin state within the fiber's quantum channel.
So the fiber is acting as a submicroscopic quantum magnetic sensor. As the biological brain fires, the magnetic field dynamically rewrites the electron spin state inside the synthetic fiber.
And the synthetic network processes that changing spin state as binary or quaternary data and transmits it out.
No batteries required. No physical electrodes prodding the tissue. It just passively piggybacks on the magnetic resonance of your own thoughts.
And it is entirely powered by the brain's own metabolic heat, utilizing the thermodynamic computing principles we discussed earlier. It represents an entirely biological, entirely integrated, structurally invisible computational overlay for the human cerebral cortex.
Okay, I need to take a deep breath here. Let's pull back and look at the sheer scale of the board Enfield has set up.
It's massive.
We have indestructible structural materials stronger than spider silk. We have massive molecular computers grown from DNA that run efficiently on ambient heat. We have synthetic Wagyu steaks written into existence by holographic lasers and invisible quantum brain interfaces woven effortlessly into our neurons.
All from the same basic architectural principle.
But every single piece of this, no matter how thermodynamically efficient it is near the Landauer limit, still requires energy. Scaling this technology from a laboratory beaker to an entire planetary civilization requires a staggering amount of power.
Naturally.
How does a Type 3 biological civilization power a world built entirely out of protein? This transitions our exploration to the fourth major pillar of Enfield's manifesto, piezoelectricity and the nuclear convergence.
The framework envisions two primary modes of power generation, starting with the ubiquitous harvesting of ambient energy. This relies on the inherent piezoelectric properties of the cross-beta architecture.
Piezoelectricity.
It's a well-understood phenomenon where a material generates an electric charge in direct response to mechanical stress. If you physically squeeze, bend, or vibrate the crystalline lattice of a piezoelectric material, the mechanical deformation forces a separation of positive and negative charges, creating a voltage.
My favorite analogy for this is squeezing a wet kitchen sponge. But instead of water dripping out of the bottom, you get a continuous trickling flow of electricity as long as you keep squeezing.
A very apt analogy. And the empirical numbers reported in the literature for these amyloid materials are highly competitive with traditional inorganic ceramics.
Do we have specific data on that?
Enfield cites a comprehensive 2024 study by Dolai et al., which demonstrated that fibrils derived from simple lysozyme and insulin proteins possess piezoelectric coefficients ranging from 24 to 42 picometers per volt.
Explain what that metric means in the real world.
It measures how much electrical charge is generated per unit of mechanical force applied. The Dolai study proved that you don't need violent impacts to generate power.
So just light taps.
Even less. They were able to generate up to 1 full volt of open circuit voltage simply by exposing a film of these biological fibrils to ambient airborne ultrasound waves. The tiny acoustic vibrations in the air were enough to warp the hydrogen bond lattice and continuously generate electricity.
So play that out at the civilizational scale. If your structural infrastructure, the massive support pillars of your buildings, the surface of your roadways, the outer hull of your spacecraft, is manufactured from these high HPD amyloid composites, the very material of your world acts as a ubiquitous, always-on generator.
Yes, the material continuously harvests mechanical energy from the chaotic environment simply by existing within it.
Like just cars driving by.
The low-frequency rumble of traffic driving over an amyloid composite bridge. The mechanical vibration of a stiff wind pressing against the side of a mile-high skyscraper.
Ocean waves.
The rhythmic, pounding impact of ocean tides crashing against a coastal seawall. Or, in an aerospace context, the continuous kinetic bombardment of the solar wind against a satellite in deep space.
That is brilliant. You completely eliminate the need for vast fields of fragile, dedicated solar panels if the entire structural mass of your civilization is constantly sweating electricity from ambient environmental vibrations.
It turns the whole city into a battery.
But Enfield doesn't stop at ambient sound and wind. The manifesto takes a massive, completely unexpected left turn into hard nuclear physics. Enfield proposed what he calls a coalescent transducer, an amyloid-based atomic-scale nuclear battery.
This is undoubtedly the most audacious cross-disciplinary synthesis in the entire framework. To grasp it, we must first understand the frustrating limitations of conventional nuclear batteries, known as beta-voltaic cells.
Right. A beta-voltaic cell is essentially a nuclear-powered solar panel. You take a lump of a radioactive isotope, something that constantly emits high-energy beta particles, which are just fast-moving electrons,
Right.
and you place it directly next to a semiconductor transducer. The isotope decays, shoots out an electron. The electron hits the semiconductor, knocks more electrons loose, and generates a direct electrical current. They are amazing because they last for decades or centuries without needing a recharge.
But they have historically suffered from abysmal energy conversion efficiencies. We are talking single-digit percentages.
Why are they so inefficient? Nuclear decay releases tremendous energy.
The primary culprit is a physical phenomenon called self-absorption. The radioactive isotope itself is a dense block of heavy matter. When an atom decays deep inside the center of that lump, the high-energy beta particle it emits has to physically travel through the rest of the radioactive lump to reach the semiconductor on the outside.
It gets stuck.
Because the material is so dense, a vast majority of the emitted electrons collide with neighboring atoms and are absorbed as useless internal heat before they ever reach the transducer. The geometry defeats the physics.
So how does the scientific community solve self-absorption?
Enfield leans on a landmark 2024 breakthrough achieved by researchers at Soochow University. They solved the self-absorption problem for inorganic chemistry by radically changing the geometry.
How do they change it?
Instead of a lump of radiation sitting next to a lump of semiconductor, they built a highly complex coordination polymer where individual radioactive isotope atoms were bound at the absolute atomic scale directly to the transducing molecules.
They integrated the fuel directly into the engine, atom by atom.
Exactly. Every single decay event was perfectly coupled to the transducer without having to travel through bulk material. By eliminating self-absorption, they achieved an astonishing 8,000-fold enhancement in energy conversion efficiency.
8,000 times more efficient.
Yeah.
That completely redefines the viability of nuclear batteries. But the Soochow University breakthrough utilized synthetic chemistry and rare earth metals. How does Enfield apply a complex inorganic physics concept to self-replicating biological protein fibers grown in a vat?
He applies it through the precision of synthetic biology and CRISPR gene editing. The framework proposes taking our foundational yeast prion sequence SUP35 and engineering its genetic code to include specific urinal binding motifs.
What exactly is a urinal binding motif?
It is a highly specialized sequence of amino acids that has evolved to have a femtomolar affinity for uranium.
Femtomolar affinity, meaning it acts like an inescapable molecular magnet for uranium atoms.
Yes. It grabs aqueous uranium ions right out of a surrounding liquid solution with unbelievable locking strength.
Okay, wait. We have to pause here. They want to intentionally bind highly radioactive uranium directly to a biological protein. My immediate reaction is that this is functionally impossible.
It sounds counterintuitive.
We all know that ionizing radiation absolutely shreds biological matter. It breaks covalent bonds, it destroys cellular tissue, it completely unravels DNA. If you bind a decaying uranium atom to a protein fiber, wouldn't the violent energy of the decay instantly obliterate the biological material holding it?
You are falling back on the assumption that biology is fragile. You have to apply the indestructibility factor we established back in Section 1.
The radiation hardening, the gamma ray resistance.
Exactly. Normal functional biology is shredded by radiation. But the tightly packed, heavily hydrogen-bonded cross-beta amyloid architecture is incredibly radiation-hardened. Gamma rays and high-energy beta particles that would completely shatter the brittle crystalline lattice of a silicon semiconductor simply wash over the nanocomposite structure of the amyloid fibril.
So the protein is simultaneously acting as the ultimate indestructible radiation shield and the power transducer.
Precisely. The manufacturing process is elegant in its simplicity. You construct an industrial vat of seawater, which naturally contains trace amounts of dissolved uranium.
Just normal seawater.
You drop in your engineered Sub-35 seed. As the seeding cascade propagates and the massive network of fibers self-assembles, the uranyl binding motifs actively pull the uranium atoms straight out of the liquid. They lock the radioactive isotopes into the core of the growing fiber at the atomic level.
The fuel is perfectly distributed at the nanometer scale.
And crucially, the uranium atom is locked in place mere angstroms away from the tyrosine pi-stacked electron conducting pathway and angstroms away from the piezoelectric hydrogen bond lattice.
So let's trace the exact physics of what happens when one of those bound uranium atoms finally decays inside the core of the fiber.
It is a masterpiece of simultaneous physics. When the atom decays, three entirely distinct energy harvesting events occur simultaneously. First, the high-energy beta electron is ejected. Because of the atomic proximity, it shoots directly into the adjacent tyrosine pi-stack channel, instantly generating a surge of direct electrical current via Hall hopping.
The beta voltaic harvest. That's one.
Second, the violent physical recoil of the decaying atom sends a kinetic shock wave rippling through the dense lattice of the fiber. The inherent piezoelectric property of the cross-beta structure instantly converts that mechanical shock wave into a secondary burst of voltage.
Harvesting the physical kick of the atom splitting. That's two.
And third, the residual localized waste heat generated by the decay event radiates outward through the liquid matrix, providing the exact thermal equilibration energy required to recharge and power the DNA prion logic gates embedded nearby.
It is harvesting a single nuclear decay event kinetically, electronically, and thermally, all at the exact same millisecond.
With zero moving parts.
There are no massive cooling towers, there are no steam turbines, and you manufacture this hyper-efficient nuclear battery by tossing some engineered bacteria into a dark vat of seawater and simply letting it grow itself into existence.
It represents a self-assembling, self-contained nuclear battery that is structurally load-bearing, meaning you could build the chassis of a rover out of it, and it can be manufactured in a bucket at room temperature. The energy density and longevity implications for civilization are simply staggering.
Okay, this brings us to the ultimate reality check.
Yeah.
We have thoroughly explored the subatomic structure, the DNA computing, the optogenetic light writing, and the coalescent nuclear batteries. But when is this actually happening?
Timeline.
Are we discussing an abstract science fiction thought experiment destined for the 22nd century? Or is this a tangible engineering roadmap? Let's examine the timeline laid out in the manifesto.
Enfield maps out a highly specific, phased development timeline that traces the evolution of this technology from current experimental lab data all the way to total civilizational integration by the year 2060.
Which is really not that far away.
And by any standard of technological forecasting, the timeline is fiercely aggressive.
Let's break it down, decade by decade, starting with the immediate future, 2025 to 2030.
In this current window, the framework projects the massive scaling of industrial fiber production. We will move away from small laboratory beakers and see the deployment of massive cyanobacterial bioreactors.
Algae, basically.
Yeah, using engineered algae to churn out high HBD amyloid fibers by the ton, using only sunlight and atmospheric carbon dioxide. During this phase, we will see the first widespread commercial deployment of the piezoelectric biosensors we discussed, integrating ambient power harvesting into everyday objects.
And the computers.
Critically, the terahertz-driven DNA computing architectures will be fully validated and stabilized at the laboratory scale.
Moving into the next decade, 2030 to 2040.
This is the decade where the technology transitions from the lab to heavy industry. The framework predicts that structural amyloid composite panels will enter rigorous aerospace qualification testing, aiming to replace aluminum and titanium in aircraft.
So planes grown from vats.
You will see the first prion DNA hybrid logic circuits operating reliably at the commercial chip scale, competing directly with high-end silicon processors. And this is the decade where the optogenetic food printers, the machines writing authentic meat microarchitecture with lasers, will move from experimental curiosities to deployed commercial pilot programs in major urban centers.
Which brings us to the era of 2040 to 2060, the era where humanity truly pushes out into deep space.
The aerospace and deep space applications are where the self-replicating biological nature of this technology becomes absolute paramount. The framework projects that by 2050, deep space exploration vehicles will utilize self-repairing amyloid hulls.
Self-repairing spaceships. Explain the mechanics of that.
Imagine a spacecraft far beyond Mars, operating in a vacuum. A micrometeorite strikes a hull and punctures it. In a traditional titanium ship, this is a catastrophic emergency requiring complex physical patches.
Right.
But the amyloid hull is an active, dormant matrix. When the micrometeorite cracks the material, the physical damage exposes the raw, broken ends of millions of amyloid fibers along the fissure.
And breaking a fiber creates a new growth point.
Exactly. The exposure of those jagged ends to the internal reservoir of liquid monomer embedded within the hull plating instantly triggers a localized seeding cascade. The fibers rapidly self-assemble across the vacuum of the crack, bridging the gap and completely healing the structural breach in minutes, utilizing the exact same thermodynamic logic that bacterial biofilms use to repair themselves in nature.
I cannot stop marveling at the implications for space colonization. Think about the current bottleneck of going to Mars. If you want to build a habitat on the Martian surface or construct a massive solar array to power a colony, you currently have to prefabricate a billion-dollar, incredibly heavy, rigid factory on Earth.
And you have to launch it.
You have to strap it to the top of a massive rocket and burn thousands of tons of highly explosive chemical fuel just to fight Earth's gravity well to get it into orbit. It is wildly inefficient. But with Enfield's paradigm, you don't send the heavy factory. You just send a seed package.
A literal test tube.
Yes. You send a test tube containing a few grams of engineered optogenetic cyanobacteria.
Yeah.
You land it in a shallow crater on Mars. You expose the bioreactor to the ambient Martian sunlight. The bacteria eats the atmospheric carbon dioxide, metabolizes the trace water ice, and you literally grow a massive structural habitat.
Or you grow an exponentially expanding, self-assembling Dyson swarm of solar harvesters.
But I have to challenge the core premise of this forecast. 2060 is practically tomorrow in civilizational infrastructure terms. Think about how many decades it took humanity to go from the invention of the first clunky silicon transistor at Bell Labs to having a sleek smartphone in every pocket. How can Enfield legitimately claim we will completely rewrite global infrastructure with a brand new material in 35 years? Isn't this timeline absurdly optimistic?
It is an aggressive timeline, without a doubt, but there is a profound structural reason driving that speed. We are undergoing a fundamental, foundational paradigm shift in the very concept of manufacturing. For the entirety of human history, from the Bronze Age to the Silicon Age, we've manufactured the world using molds.
Molds require infrastructure.
Molds require devastatingly massive infrastructure. To build a modern silicon chip, you need a $20 billion fabrication plant. You require global mining operations to extract rare earth metals. You need high-temperature smelting furnaces. You need highly toxic photolithography chemicals. You need supply chains that stretch across a dozen countries. The scaling of mold-based manufacturing is severely constrained by physical capital and human logistics.
But this biological technology,
This technology abandons the mold entirely. It operates entirely on the paradigm of seeds. The massive billion-dollar manufacturing factory is replaced by a single microscopic photosynthesizing microorganism floating in a glass bioreactor. The raw materials are no longer mined rare earth metals. They're simply ambient carbon dioxide, water, and sunlight.
which are infinitely abundant everywhere on the planet.
Because the factory is the organism itself, the marginal cost of creating high-tensile structural materials, quantum computers, and nuclear batteries approaches absolute zero. When your entire industrial base is composed of self-replicating biology that runs free on sunlight and heat, the traditional economic scaling laws dissolve completely. You are no longer limited by how much capital you can raise to build a factory, or how fast ships can cross the ocean with raw materials. You are limited solely by the exponential biological growth rate of the bacteria in the vat.
which means the speed of integration will be blinding. So by 2060 and beyond, Enfield is actively envisioning full civilizational integration, a society where our physical architecture, our high-speed quantum computing, our energy grid, and our internal medicine are all completely unified into a single, highly programmable biological substance.
He classifies this theoretical future state as a Type 3 biological node. It describes a civilization whose very bodies and whose surrounding manufactured environments are no longer distinct from one another. The organism is the civilizational infrastructure.
Okay, we are coming to the close of our deep dive, but we absolutely must address the elephant in the room. We briefly touched on it at the very beginning, but the sheer visceral fear factor surrounding this technology is immense. If we intentionally engineer vast vats of indestructible, self-replicating prions, what happens if there is a containment breach? What happens if they get loose? Are we accidentally engineering the ultimate unstoppable biological weapon? What if these hyper-tough fibers decide to initiate a seeding cascade inside the local municipal water supply, or worse, inside the neural tissue of the human population?
It is the most critical safety question raised by the entire framework, and Enfield addresses the containment protocols head-on. The terrifying pathogenic dimension of mammalian prion disease, the mad cow scenario, cannot be ignored or minimized. But the resolution found within the synthetic engineering toolkit is clean, strict, and entirely molecular.
Walk us through the safety mechanism.
The proteins being utilized for all of these advanced computational and structural applications, the sequences being grown in the vats, are not the mammalian PRP prion that misfolds and causes human neurodegenerative disease.
So what exactly are they using?
As we discussed, they are utilizing specific yeast prion determinants, most notably the SUP35 sequence, alongside completely synthetic computer-designed peptides that have no analog in nature. These engineered proteins share the architectural principle of self-assembly, the ability to form the rigid cross-beta tower, but they strictly and fundamentally exclude the pathogenic mechanism of mammalian diseases.
So it's similar to how modern medicine uses the concept of a virus to deliver a life-saving genetic therapy. You are borrowing the incredibly efficient delivery mechanism, the virus's mechanical ability to dock with a cell and inject a payload, but before you use it, you completely strip out all of the malicious viral DNA that actually causes a cold or debilitating disease. In this framework, they are utilizing the physical mechanism of the templating cascade, but they are absolutely not importing the pathology.
Exactly. The synthetic engineering borrows the elegant architecture of the fold, not the disease. The thermodynamics of the engineered sequence are locked. The SUP35 seed only has the physical geometry to recognize and template its exact specific sequence. That specific fold has zero physical mechanism and zero thermodynamic incentive to interact with the natural human proteome in a pathogenic, disease-causing manner. In fact, the original 2003 PNAS study that first demonstrated growing prion nanowires in a lab explicitly confirmed through exhaustive testing that the SUP35 yeast prion is entirely non-infectious to mammalian biology.
That is a crucial distinction. Okay, let's distill this down for you, the listener. What is your fundamental takeaway from navigating this incredibly dense, paradigm-shifting framework today? The next time you open a news app and see a headline about a breakthrough in quantum computing architecture, or a massive new discovery in synthetic biology, or a newly engineered material for deep space solar panels, I want you to stop and realize that the rigid academic barriers between those disciplines are rapidly dissolving.
For over a century, the scientific community has treated biology as something squishy, wet, fragile, and entirely distinct from the hard, rigid, mathematical world of structural engineering and quantum electronics. Enfield's manifesto proves, with rigorous physical chemistry, that this is a false dichotomy. Biology is not merely the study of life. Biology is the next iteration of hardware. It is the most advanced, energy-efficient, atomically precise manufacturing platform in the universe, and we are finally, haltingly, learning how to write the source code for it.
And that leads me to a final, slightly provocative thought I want to leave you with. It builds on a concept buried at the very deepest theoretical layers of Enfield's theory, the holographic information geometry.
Ah, the application of the Bekenstein-Hawking connection to biological fibers?
Yes, it is a concept borrowed from the physics of black holes. The theory suggests that in an amyloid fiber, the complete three-dimensional blueprint of the entire massive structure is mathematically written entirely on its two-dimensional surface interface. The 2D surface geometry at the end of the fiber is the memory storage, it is the executable program, and it is the physical structure all operating simultaneously at once. The tiny part flawlessly contains the massive whole.
That's a staggering proposition.
And if that is true, If we can physically write that geometry into solid matter using nothing but pulses of holographic light,
Yeah.
if we can weave synthetic quantum processors directly into the wet neural matrix of our own brains, reading our deepest thoughts through the microscopic magnetic resonance of our own pulse.
The boundary completely vanishes.
Exactly. At what specific point does the hard line between living organism and engineered machine completely disappear? If our technology becomes something that physically grows inside ourselves, something that runs on our own body heat and shares our biological matrix, when does our technology stop being something we actively use and start being something we fundamentally are?
It is a profound, almost philosophical transition point for the species. We are no longer simply building tools to manipulate our environment. We are physically expanding the definition of the human organism.
At the very start of this deep dive, we talked about prions as biological errors. We like the world to be neatly categorized. We like a clean, sharp line between the soft biology of a human being and the hard machinery of a computer. But as we look toward the timeline of 2060, that categorization is breaking down. We are rapidly walking into a world where the concrete highway, the supercomputer, and the organic brain are all woven from the exact same indestructible thread. Thank you for taking the plunge into this total application framework with us. Keep questioning the lines you see in the world around you.
So I want you to take a look at the device you're using to listen to us right now. Maybe it's your phone or maybe a laptop.
Right, whatever's in front of you.
Exactly. Just run your finger over the casing for a second. Inside that shell is this insane labyrinth of silicon, copper, and all these rare earth metals.
Which all had to come from somewhere, right?
Yeah, I mean, every single microchip, every battery cell in that device was literally ripped out of the earth by heavy machinery. It was shipped across oceans, melted down in blast furnaces at like thousands of degrees.
And forged in factories that consume the power of small cities.
Exactly. That is the fundamental premise of human infrastructure. We build by extracting, burning, and forcing dead matter into useful shapes.
Which, if you think about it, is an incredibly fragile and just, well, brute force way to build a civilization.
It really is.
I mean, we spend more energy fighting the natural entropy of our materials than we do actually utilizing them.
Right, and here is the hook for today's deep dive. What if I told you that your next phone, or the car you drive, or even the building you're sitting in, won't be built in a factory at all?
It's a massive paradigm shift.
Totally. What if it's grown in a vat of water at room temperature using sunlight out of the exact same proteins that cause mad cow disease?
I know it sounds completely absurd when you frame it that way.
It sounds like a sci-fi horror movie.
It really does. But we are looking at a massive synthesis of peer-reviewed literature today. This spans over 2 decades, and it culminates in Zachary David Enfield's 2026 framework.
And this proves it isn't just theoretical anymore, right?
Exactly. It proves this is engineered reality. We are talking about taking the most feared structural anomaly in biology, the prion, and completely reprogramming it.
Into a building material.
Yes, a unified architectural material, a material that computes, generates its own nuclear power, and is theoretically capable of building Dyson swarms around stars.
Okay, I want to walk through this carefully because the sheer scale of the documents we're looking at today is just staggering.
It covers a lot of ground.
It bridges quantum physics, synthetic biology, macroengineering. I mean, let's start at the absolute foundation, the material itself.
That's the best place to start.
Because when we say the word prion, most people, myself included, immediately think of biological errors, brain-wasting diseases.
Right, mad cow, things like that.
Yeah, we think of something tiny, biological, and incredibly dangerous. So what are we actually looking at when we zoom in on the physical structure of a prion fiber?
Well, we're looking at what structural biologists call the cross-beta architecture.
Cross-beta.
Okay. Yeah. Let's strip away the disease aspect for a minute and just look at the geometry. Have you ever played with a slinky?
Oh, sure, yeah.
Imagine that slinky, but instead of a loose coil, it's tightly flattened out into this zigzag pattern.
Okay, I'm picturing it.
That zigzag is called a beta strand. Now, stack thousands of those flat zigzags directly on top of each other.
Like pancakes.
Kind of, but you bind them together with a really dense network of hydrogen bonds. That entire stack creates a beta sheet.
Okay, so it's almost like a microscopic deck of cards, where every single card is superglued to the one above and below it.
That's actually a really helpful starting point. But a deck of glued cards is stiff, right? It's brittle.
Yeah, if you bend it too far, it just snaps.
Exactly. The cross-beta architecture solves this brittleness by alternating those rigid hydrogen-bonded layers with flexible amino acid side chains.
So, wait, meaning there are little molecular shock absorbers built between the rigid plates?
That's exactly what they are. Think of it like the suspension system in a heavy-duty truck.
Oh, okay.
You had this incredibly stiff steel frame that's your hydrogen-bonded beta sheet, and that provides immense tensile strength.
But the truck doesn't shatter when it hits a pothole.
Right, because when the material takes an impact, those amino acid side chains flex and dissipate the kinetic energy as heat.
That is so elegant.
It's a naturally occurring nanocomposite. I mean, nature already uses this kind of design logic in materials like nacre, the mother of pearl inside abalone shells.
Oh, yeah, that stuff is super tough.
It is, and it uses a similar principle to stop cracks from propagating.
Mm-hmm.
But here, with the prion fiber, the architecture is perfected down at the 10-nanometer scale.
And when you look at the mechanical testing data in the literature provided, the results are just baffling.
They really challenge our assumptions about biology.
Seriously. Because we usually treat spider silk as the holy grail of biomaterials, right? Like every pop science article for the last 10 years has been about how spider silk is stronger than steel.
It's the classic comparison, yeah.
But this engineered polymeric amyloid, this synthetic prion material, it actually beats spider silk in toughness.
Significantly.
The data shows it hitting 160 megajoules per cubic meter.
Yeah, and we should probably define toughness here because people often confuse it with strength.
Right, they aren't the same thing in physics.
Not at all. Think about a diamond. If you pull on a diamond, it has immense strength. It won't stretch.
Yeah.
But if you hit a diamond with a hammer...
It just shatters into dust.
Exactly. Because it has very low toughness. Toughness is the total amount of mechanical energy a material can absorb before it catastrophically fails.
Okay, so it's about energy absorption.
Yes. This cross-beta amyloid absorbs a staggering amount of energy. It bends, stretches, and recovers, all while maintaining a mass that is incredibly 1/8 the density of structural steel.
1/8 the density, and it's the chemical indestructibility that I find even crazier than the physical toughness.
The chemical stability is where it gets really wild.
Because our bodies are full of proteins, and proteins are notoriously fragile. I mean, if you get a high fever, your proteins literally start to denature.
Right, they lose their shape and stop working.
But these specific prion fibers, the research shows scientists boiling them in concentrated sodium hydroxide.
Which is incredibly costly.
Yeah, they soak them in pure formic acid, they blast them with massive doses of gamma radiation.
Doses that would instantly melt a standard silicon logic chip.
Or just vaporize copper wiring entirely, and the amyloid just sits there, unfazed. How is that even possible?
It goes back to the sheer density of those hydrogen bonds. It creates what we call a thermodynamic sink.
A thermodynamic sink? What does that mean, practically?
It means it is the most energetically stable shape a protein chain can possibly take in the universe.
Wow.
To pull it apart requires more energy than the chemical bonds of the individual atoms can sometimes withstand. You'd break the atoms apart before you break the shape.
That is mind-blowing.
But the true genius of Enkield's framework isn't just that the material is indestructible, it's that it's tunable.
Tunable.
Engineers refer to this as the hydrogen bond density dial, or the HBD dial.
Oh, right, I saw this in the text. Which means we can change the physical properties of the fiber just by tweaking the genetic code of the protein before it grows, right?
Exactly. By altering the specific sequence of amino acids, you dictate how tightly those zigzags pack together.
So you're adjusting the glue?
Yeah. If you engineer a sequence with incredibly tight residue packing, you crank the HBD dial all the way up. The resulting fiber behaves like a structural steel beam for an aircraft.
Okay, super rigid.
But if you introduce slightly bulky or polar amino acids into the core, you essentially lower the hydrogen bond density.
And what does that do?
You get a fragile, dynamic fiber that easily fragments.
And I want to pause on that fragmentation point, actually, because earlier I kept thinking of this material like an indestructible Lego brick.
Which is a common way to visualize it.
But that analogy is incomplete, isn't it? Because a Lego brick is static. You snap two together, you just have a bigger piece of plastic. But this material is, well, it's almost alive.
Self-replicating is the precise term.
Right, self-replicating, because if you take a steel beam and snap it in half, you just have a broken beam. It's garbage.
Which is useless, yeah.
But if you snap one of these dynamic prion fibers in half, you've just created two brand new growth points. Every broken end is an exposed template.
Hmm.
If you drop that broken piece into a vat of liquid monomer, which is just raw, unfolded proteins floating in water, that broken tip physically forces the free-floating proteins to fold into its exact shape.
It's an unstoppable geometric coercion.
It's like dropping a single ice crystal into super cold water, and suddenly the whole vat just freezes into a solid block.
The mechanism is called templated seeding.
Templated seeding.
Yeah, the surface chemistry of the fiber tip lowers the activation energy required for the free-floating monomers to fold.
So it makes it easier for them to change shape,
It essentially forces them into adopting the cross-beta shape, adding them to the chain one by one.
which means your manufacturing process requires basically zero external energy.
Zero. No heat, no smelting, no massive robotic assembly lines.
That's zero.
You just need a gentle mechanical mixer to occasionally break the growing fibers, which creates more exposed ends, leading to exponential replication.
So the manufacturing cost approaches the literal cost of the raw protein soup.
It's basically free to grow once you have the starter seed.
Okay, but I have to push back hard here.
Go ahead.
Because you are calmly describing a scenario that sounds like an apocalyptic sci-fi movie.
I know how it sounds, believe me.
We have an indestructible, highly stable protein that forcefully corrupts other proteins into its exact shape, spreading exponentially just by touching them.
Yes.
You are describing the exact mechanism of mad cow disease. You are describing how a single misfolded protein literally eats a human brain. Why on earth would we want to mass produce this and build our homes and our computers out of it?
It is a vital question, and it's completely understandable to be alarmed. But the framework addresses containment as the absolute baseline of the technology.
Okay, so how is it safe?
The key distinction is the specific protein sequence being used.
Hmm.
Mammalian prion diseases like mad cow or Creutzfeldt-Jakob disease are caused by the misfolding of a very specific protein in our bodies called PRP.
Right, the one that naturally exists in our brains.
Exactly. That specific protein sequence interacts with our neural tissue.
So the engineers aren't using PRP at all.
Not at all. They utilize harmless yeast prion determinants, primarily a protein called sub-35, or they design entirely synthetic, non-biological sequences from scratch.
Let me see if I have this right. It's like we realized a bank robber used a really incredible getaway car.
Okay, I like this analogy.
Instead of hiring the bank robber, we just stole the blueprints for his car engine and put it in a tractor.
Yes.
We're taking the pure mechanical horsepower of the self-assembly, but we completely threw away the genetic sequence that knows how to interact with human brain cells.
A very apt way to visualize it. A yeast prion sequence has absolutely no biological pathway to infect human tissue.
So it's totally inert to us.
Right. We are harnessing the physics of the assembly mechanism while completely discarding the biological pathology.
Okay. I can accept that the soup won't eat my brain.
It definitely won't.
But that leads to an immense engineering problem. We have a vat of harmless, self-assembling, indestructible wire. How do we pellet what to build?
Control.
Yeah. If it just naturally grows in every direction the moment a monomer touches a seed, you don't get a comparator chip or a structural beam. You just get a giant, tangled block of biological concrete.
Which isn't very useful.
Not at all. How do we draw a deliberate shape with this stuff?
This is where we see the convergence of biology and classical microchip manufacturing.
Okay, how so?
The framework details a process called prion optogenetic lithography.
Prion optogenetic lithography.
Yes. We are essentially giving the protein a set of breaks, and the only way to release those breaks is with light.
Optogenetics. I've seen this used in neuroscience, where researchers flash lasers into a mouse's brain to activate specific neurons.
Exactly the same field.
But how does that apply to a structural material? How do you put brakes on a building material?
Researchers take the DNA sequence for our harmless prion domain, and they fuse it to an optogenetic domain.
Which is what, exactly?
It's a light-sensitive protein derived from plants or bacteria, like Cry2 or LOV2. Think of this light-sensitive domain as a microscopic cage.
So they genetically stitch a cage onto the prion.
Correct. We create a chimeric protein, two distinct parts joined together.
Got it.
Now, as long as this chimeric protein is kept in the dark, the optogenetic cage stays tightly wrapped around the prion domain.
So it can't interact with anything.
Right. The protein is locked in an inert state. It cannot template, it cannot seed, it cannot grow. You can have a massive 10,000-gallon vat of this liquid protein, and it will remain a liquid indefinitely.
Until you turn on the lights.
Exactly.
But we aren't just flipping a light switch in the room, right? Because we need nanoscale precision to draw a microchip.
Right. A light bulb would just turn the whole VAD solid.
Right.
We used a 2-photon femtosecond laser.
A femtosecond laser? That sounds fast.
It is a highly specialized laser that fires pulses of light lasting quadrillionths of a second.
Quadrillionths.
And because it requires 2 photons hitting the exact same molecule at the exact same time to trigger a reaction, the focal point of the laser is incredibly tiny.
How tiny?
Down to the nanometer scale. Furthermore, the wavelength of light passes harmlessly through the rest of the liquid. The energy is only absorbed right at that microscopic focal point.
So when that specific pinprick of laser light hits the chimeric protein in the dark vat,
The ontogenetic cage absorbs the light energy and physically changes shape.
it pops open.
The cage springs open. The prion domain is suddenly exposed, and in a fraction of a second, it snaps into its cross-beta architecture and bonds to its neighbor.
It's basically a 3D printer, but completely inverted.
That's a great way to put it.
Because in a normal resin 3D printer, a laser melts or cures a toxic liquid plastic layer by layer.
Yes, curing dead matter.
But here, the laser is just acting as a traffic light. It's shining a green light onto a specific microscopic coordinate, telling the biological seeds right in that spot, okay, you have permission to grow now.
And it traces the blueprint in 3 dimensions.
And the control is absolute, right? I read about the 2025 paper in the source material.
The Nature Communications paper on the phobeat system, yes.
Yeah, that one. It demonstrated that this is a fully modular light switch.
Completely modular. You can use blue light to trigger the assembly, draw a microscopic structural truss.
And then flash it with red light to instantly close the cages.
Exactly. Capping the ends of the fibers so they stop growing entirely. You define the exact physical boundaries of the architecture using nothing but photons.
That is wild. The applications laid out in the research for this are just, they feel like magic.
They blur the line between tool and medicine, certainly.
Let's look at the medical side because that really highlights what you just said. There is a scenario in the text where a doctor uses a viral vector, an adeno-associated virus, or AAV, to inject the genetic instructions for this light-activated material directly into a patient's tumor.
Right. The virus acts as a harmless delivery vehicle.
Okay.
It inserts the code into the tumor cells, which then start manufacturing the caged inert proteins.
Just building up the liquid inside the tumor.
Right. And because the patient is opaque, it's completely dark inside the tumor. Nothing happens. The proteins just pool there.
Then the oncologist inserts a tiny fiber optic needle directly into the tumor and pulses the specific laser pattern.
Shining the blueprint.
Yes. Suddenly, this indestructible, rigid amyloid scaffold begins violently growing through the tumor cells.
Tearing it apart from the inside.
But it's not just a physical stake through the heart of the cancer, which is crazy enough. The framework notes that these specific fibers are piezoelectric.
Let's unpack that term for the audience.
Please do.
Piezoelectricity is the ability of certain materials to generate an electrical charge in response to applied mechanical stress.
So pressure equals electricity.
Basically, if you squeeze or vibrate a piezoelectric crystal, it produces voltage.
So the doctor has grown a microscopic voltage-generating crystal lattice inside the tumor. How do they trigger it without cutting the patient open?
From the outside, using a standard ultrasound machine.
Just sound waves.
Yes. The harmless sound waves pass through the patient's body and physically vibrate the newly grown amyloid scaffold.
Wow.
That vibration causes the fibers to generate localized electrical spikes.
Inside the cancer cells.
Yes. And those electrical spikes strip electrons from the surrounding water and oxygen, creating reactive oxygen species.
Which are highly toxic, right?
Highly toxic oxidative molecules. It essentially electrocutes the cancer cells from the inside out, selectively destroying the tumor while leaving the healthy surrounding tissue totally untouched.
And what happens to the scaffold after the tumor is dead?
The doctor simply alters the light frequency, the scaffold destabilizes, and it dissolves harmlessly back into basic amino acids.
It's a localized, self-assembling, electrically powered chemotherapy that leaves zero toxic trace.
It's incredibly elegant.
And if you scale that exact same optogenetic lithography up, you get the manufacturing applications.
Which are just as disruptive.
The literature talks about printing massive lattices for synthetic meat or growing a replacement femur for a patient by projecting a holographic laser blueprint into a bioreactor.
Right.
You aren't printing dead matter, you are orchestrating growth.
It's writing architecture into existence.
Writing architecture, I love that.
Yeah. But we have to look closer at what this material can do physically, because it isn't just tough. And it isn't just piezoelectric. The framework highlights that the cross-beta core of the amyloid is intrinsically conductive.
Right. The text mentions something called tyrosine pi stacking. I need you to break that down because I feel like this is the bridge to the next massive leap in this framework.
It absolutely is.
How does a protein conduct electricity like a copper wire?
Well, tyrosine is a specific amino acid with a ring-shaped chemical structure.
Okay, picture a ring.
When these proteins fold into their tight, zigzag beta sheets, those tyrosine rings end up stacked directly on top of each other, incredibly close together.
Like a stack of donuts?
Sure, a stack of donuts. Now, in chemistry, these rings have clouds of electrons hovering above and below them called pi orbitals.
Okay.
Because the rings are packed so tightly in the prion fiber, those electron clouds overlap, creating a continuous unbroken tunnel, a pi stacking highway.
So the electrons can just slide right down the stack.
Exactly. Electrons can flow right down the center of the fiber.
Okay, so let's summarize what we have so far for the listener. We have a material that is stronger than spider silk, survives acid and radiation, can be drawn into any 3D shape using lasers, and natively conducts electricity.
That's the foundation, yes.
What happens when you use lasers to draw a microscopic three-dimensional wiring diagram using this conductive, indestructible material?
You build a computer,
A biological computer.
but not a computer as we currently understand it. This brings us to the 8-layer unified biological computer.
This part of the deep dive blew my mind.
It's incredibly dense.
Because it redefines what computation actually is. It's not just ones and zeros on a hard drive. Let's walk through these layers starting at the absolute bottom. Layer 1, the electron layer.
Okay, let's start there.
We just talked about the tyrosine highway, but the text says this layer uses quantum tunneling and vibronic coherence. What does that actually mean in a biological wire?
Well, in a traditional copper wire, electrons bump along like cars in traffic, losing energy as heat.
Right, resistance.
But at the angstrom scale, inside this highly ordered amyloid crystal, we see quantum effects. The framework draws a direct parallel to the FMO complex.
The Fenner-Matthews-Olsen complex, right,
Yeah.
from photosynthesis.
Yes, exactly. For a long time, physicists couldn't figure out how plants transfer solar energy from their surface to their reaction centers with 99% efficiency. It seemed physically impossible.
Because there should be loss along the way.
Right. It turns out the biological structure in the leaf creates a state of quantum superposition.
Whoa!
The energy particle explores all possible paths simultaneously and instantly chooses the most efficient one.
But quantum coherence usually requires temperatures near absolute zero, doesn't it? Like the quantum computers Google is building.
Usually, yes. Otherwise, the random vibrations of the surrounding atoms, the thermal noise, destroys the quantum state.
So how does the plant, or our prion wire, do it at room temperature?
Through vibronic coherence.
Vibronic coherence.
The cross-beta architecture is so rigidly ordered that its molecular vibrations as phonons act as a synchronized bath.
What does that mean, a synchronized bath?
Instead of random thermal noise disrupting the electron, the vibration of the protein actually sings in harmony with the electron's quantum state. It shields it and pushes it along the tyrosine highway.
The wire itself is a quantum-enabled environment dictated purely by its physical geometry.
Exactly. The shape creates the quantum shielding.
That is wild. Okay, moving up the stack, let's jump to layer 3, which the framework calls the RNA execution layer.
The dynamic logic layer.
Yeah, because we always think of DNA as the hard drive of biology. What is RNA doing here?
RNA is acting as the dynamic logic gates and the analog to digital converters. Within the liquid environment flowing around the amyloid nanowires, researchers encode RNA aptamers and riboswitches.
I need an ELI5 on riboswitches. How does a strand of RNA act like a computer chip?
Think of a riboswitch as a biological thermostat combined with a switch. It's a folded piece of RNA that is designed to change its shape only when it binds to a very specific target.
Like what kind of target?
It's a specific chemical pollutant or a specific ion. When the target binds, the RNA snaps into a new shape, which physically exposes a new sequence that can trigger a downstream chemical reaction or interact with the conductive fiber.
Oh, I see.
It is constantly sampling the continuous analog chemical environment and converting that information into discrete digital on-off signals for the computer to process.
It's the working memory and the environmental sensors wrapped into one.
Exactly.
But the heavy lifting, the mass data storage, happens at layer 6, the GNA and terahertz layer. And this is a massive upgrade over silicon binary logic.
A monumental upgrade. Silicon computing is trapped in binary. The transistor is either open or closed, a 1 or a 0.
Right.
DNA fundamentally operates on a quaternary system. You have 4 states: adenine, cytosine, thymine, and guanine.
So moving from base 2 to base 4 logic.
It exponentially increases your data density.
Yeah.
A single gram of DNA can theoretically store hundreds of petabytes of data.
But the Achilles heel of DNA computing has always been speed, hasn't it?
That has been the major bottleneck, yes.
Like, you can store the whole internet in a test tube, but if you want to search for a file, you have to wait for chemical kinetics.
Which is agonizingly slow.
You have to wait for enzymes to literally bump into the DNA, unzip it, read it, and rebind it. It takes hours.
Which is utterly useless for real-time computation.
Right, so how do they fix it?
This is where the framework introduces the terahertz clock signal. We bypass chemical enzymes entirely and use pure physics.
Pure physics to read DNA, how?
The hydrogen bonds holding the 2 strands of the DNA double helix together have specific resonant frequencies.
Like a tuning fork.
Exactly. If you blast the DNA with terahertz radiation, which sits between microwaves and infrared light on the spectrum, and you tune it to that exact resonant frequency,
You can vibrate it apart.
you forcefully vibrate those bonds until they snap open. You can unzip the DNA, alter its state, and let it snap back together in picoseconds.
A trillionth of a second.
Yeah.
So you replace the slow chemical scissors with a hyperfast electromagnetic tuning fork. You transition DNA data storage from slow chemistry to ultrafast physics.
Exactly. You're bringing biological data speeds into the realm of modern microprocessors.
That's incredible.
And integrating all of this together is layer 7, the photonic layer.
The optical layer, right?
We can physically attach quantum dots, which are nanoscale semiconductor particles that emit single photons of light at precise coordinates along our amyloid fibers.
But the fiber isn't just a straight line, is it? The structure has a natural twist.
Yes, it is a helical chiral structure. And because of that physical twist, it acts as a chiral waveguide.
What does that do to the light?
If a quantum dot fires a photon down the fiber, the physical twist of the protein naturally polarizes the light, routing left-handed and right-handed photons in different directions.
Oh, wow.
The amyloid wire natively functions as a beam splitter for a room-temperature optical quantum computer.
Okay, I am following the mechanics here. But to understand why we actually need this, why we'd replace our silicon data centers with vats of laser pulse protein, we have to talk about layer 5.
Thermodynamic computing.
Yes. We need to talk about heat and the Landauer principle.
The Landauer limit is the invisible wall that modern computing is currently crashing into.
Why are we crashing into it?
Well, in 1961, physicist Rolf Landauer proved a fundamental law of thermodynamics regarding information. He showed that any irreversible computation specifically, the act of erasing a bit of information, must dissipate a minimum non-zero amount of heat into the environment.
Because you're decreasing entropy locally by erasing the data, so the universe demands an entropy tax in the form of heat somewhere else.
That's the core of it, yes. Traditional silicon logic gates are irreversible.
Give me an example.
Take an ND gate. 2 input bits go in and 1 output bit comes out. The gate physically destroyed 1 bit of information to perform the logic.
And that destruction generates heat.
Exactly. When you have billions of transistors doing this billions of times a second, your microchip gets blistering hot.
This is why AI data centers consume rivers of fresh water and require gigawatts of cooling infrastructure.
They are fighting a losing battle against the laws of thermodynamics.
But this biological 8-layer amyloid computer doesn't fight thermodynamics. How does it bypass the Landauer limit?
By utilizing conformational cascades, which are forms of reversible computing.
Reversible computing, meaning no data is destroyed.
Right. When a prenon logic gate shifts from a 0 to a 1, it doesn't destroy the previous input state. It geometrically copies it.
Like how?
Think of a row of dominoes falling. The first domino doesn't disappear when it hits the second. It physically transfers its state forward.
Ah, okay.
The upstream amyloid fiber retains its physical conformation, while the downstream fiber reads and transmits it. Because no information is permanently erased from the physical geometry, the system doesn't have to pay the entropy tax.
So it generates no heat?
It operates near the theoretical floor of zero heat computation.
Okay, I have to stop you there.
What's the issue?
Because I read the dissenting papers in the source material, and there is a glaring, massive contradiction here.
Oh, I know where you're going with this.
You just said it operates with zero heat, but earlier you explained that the terahertz clock signal is what unzips the DNA for processing.
I did say that.
Well, physicists point out that terahertz radiation is almost entirely absorbed by liquid water. Water is basically a brick wall to terahertz frequencies. It absorbs the waves and immediately turns them into heat.
That is correct.
And this entire biological computer, the DNA, the RNA, the proteins, only functions if it is submerged in a wet, aqueous environment.
Yes, it needs water.
So if you blast this system with a terahertz clock, the water absorbs the signal, turns it into heat, and essentially boils the computer alive. The signal never even reaches the DNA.
It is an incredibly astute pushback. And for years, it was considered the fatal flaw of TRT's biological interfacing,
Right. So how is it not a flaw?
because it was completely inverted by a 2025 paper in Nature by Song and Kian from Caltech.
Okay, what did they find?
The researchers proved that the heat isn't destroying the system. The heat is the battery.
Wait, I'm trying to wrap my head around that. How can the flaw be the fuel? If the water boils, the proteins denature, right?
We aren't boiling the water. We are inducing microscale thermal gradients.
Okay.
Song and Kian were working with enzyme-free DNA logic circuits. They found that once a DNA circuit reaches equilibrium, meaning it has completed its calculation and is chemically exhausted, it stops working.
Like a dead battery.
Exactly. To reset the circuit, to push it back into an out-of-equilibrium state so we can compute again, it requires an influx of energy
So where does the energy come from?
from thermal ramps. The heat generated by the water absorbing the terahertz pulse creates localized spikes in temperature.
Oh, wow.
Those specific thermal fluctuations physically agitate the DNA, resetting the base pairs and pushing the logic gates back to their high-energy starting positions.
So the thermal dissipation from the water isn't a bug that breaks the machine?
No, it is the exact physical mechanism that recharges the biological circuit.
Let me make sure I'm hearing this right. We are blasting a signal, the water gets in the way and creates a blast of heat noise, and the computer is designed to harvest that random thermal noise to wind its own clock springs back up.
You're hitting on the fundamental shift in paradigm here. Silicon computing tries to isolate itself from environmental noise.
Right, and wants a sterile, perfectly cold vacuum.
Biological computing surfs on the environmental noise.
Yeah.
It harvests the random Brownian motion and thermal gradients of its environment as its computational engine. It is a heat-rechargeable computing substrate.
It's elegant. It takes the chaos of the natural world and weaponizes it for logic.
Exactly.
But it brings up a practical reality. We have an infinitely scalable self-assembling computer that operates at the thermodynamic limit of physics. But macro-scale infrastructure requires macro-scale power.
Yes, computing is only half the battle.
Right. If we are replacing our silicon infrastructure with amyloid vats, we can't exactly plug our biological computers into a coal plant. Let's explore how this material powers itself.
To understand the energy harvesting architecture, we have to look at the historical failures of nuclear batteries, specifically beta voltaic cells.
Beta voltaics, these are batteries that run on radioactive decay, right? Not fission, just the natural breakdown of isotopes.
Correct. In a traditional beta voltaic battery, you take a chunk of a radioactive isotope, like nickel-63 or tritium, and you place it next to a semiconductor transducer.
Okay.
As the isotope naturally decays over decades, it spits out high-energy electrons, beta particles. Those electrons hit the semiconductor, knock other electrons loose, and create an electrical current. It's a battery that lasts for 50 years.
Sounds like the perfect power source. Why aren't they in our phones right now?
Because of a physical limitation called self-absorption.
Self-absorption.
There is a physical gap, even if it's microscopic, between the chunk of radioactive material and the semiconductor capturing the energy.
Right, they are two separate pieces.
As the beta particle travels out of the isotope and across that gap, it crashes into other atoms. It loses almost all of its kinetic energy as useless heat before it ever reaches the semiconductor.
Oh, I see.
Because of this gap, state-of-the-art beta voltaics achieve, at best, a single-digit percentage of energy conversion efficiency. They produce microwatts.
Enough to run a pacemaker, maybe, but not much else.
Exactly.
So how does the amyloid framework solve the gap problem?
By eliminating the concept of components entirely. In 2024, researchers at Soochow University published a breakthrough in Nature demonstrating the coalescent transducer.
The coalescent transducer, what does that do?
Instead of putting the radioactive fuel next to the solar panel, they chemically bonded the radioactive isotope directly into the molecular lattice of the transducer itself.
So the fuel and the engine are literally the exact same molecule.
Yes. By coupling the emitter and the transducer at the angstrom scale, they bypass the self-absorption gap entirely.
What kind of efficiency jump are we talking about?
They achieved an 8,000-fold enhancement in energy conversion efficiency.
8,000 times.
Mhm.
Because the atomic explosion, well, the decay event is happening literally inside the material that captures the energy. Nothing is lost to the void.
Precisely. Now, Enfield's framework takes this inorganic chemistry principle and applies it to our synthetic biological scaffold using CRISPR.
How do you CRISPR a nuclear battery?
By base editing the genetic sequence of the cyanobacteria that produces our prion monomers, engineers insert highly specific urinal binding motifs into the protein chain.
Urinal binding motifs.
These are molecular traps with femtomolar affinity, meaning they are so incredibly sensitive they can pluck a single specific atom out of a billion others.
Wow.
You can run seawater, which naturally contains trace amounts of uranium, through the growing amyloid vat.
And it just filters it.
The protein scaffold will actively pull the uranium atoms out of the water and lock them at precise angstrom-level coordinates directly inside the conductive fiber.
Just to reassure anyone listening who is immediately imagining a biological Chernobyl, we are not talking about a nuclear fission chain reaction here, right? We aren't splitting atoms.
No, not at all. We are simply capturing the ambient, slow, natural decay of the isotope.
Okay, phew.
But what makes the amyloid scaffold so revolutionary is its trimodal harvesting capability.
Trimodal. So it captures energy in 3 ways.
A traditional beta voltaic only captures the emitted electron. The coalescent amyloid transducer captures 3 distinct forms of energy from a single decaying atom simultaneously.
Walk me through the 3 channels.
Channel 1 is the traditional beta voltaic route. The atom decays, shoots out a beta particle, and that electron is channeled directly down our tyrosine pi stacking highway as electrical current.
Got it. The electron highway.
Channel 2 is the piezoelectric route.
The vibration harvesting we talked about?
Right. Think of firing a heavy rifle. The bullet goes forward, but the gun kicks back into your shoulder.
Right. Recoil.
When the radioactive atom spits out a particle, the nucleus violently recoils. It sends a physical shock wave, a phonon cascade, rippling through the rigid cross-beta crystal lattice.
Oh.
Because our amyloid is highly piezoelectric, it absorbs that mechanical shock wave and instantly converts the physical vibration into a voltage spike.
So it captures the electron and the physical recoil of the atom. That's 2. What's the 3rd channel?
The thermal gradient. The sheer violence of the decay event generates localized heat right at the binding site.
Okay.
This creates a microscopic temperature difference between the hot core of the fiber and the cooler surrounding water. The material acts as a thermoelectric generator, harvesting that temperature difference into additional current.
Electron, vibration, heat, every possible drop of entropy is captured.
Exactly.
And this vibration harvesting piezoelectric property isn't just limited to atomic recoil. The framework expands this to massive mechanical systems.
The macro applications are staggering.
Give me an example.
Imagine a deep-sea submarine. Instead of building the hull out of titanium, you grow a meter-thick shell of this cross-beta amyloid composite.
Okay.
At 600 meters down, the ocean exerts immense, continuous hydrostatic pressure on the hull. The piezoelectric amyloid matrix literally harvests that mechanical stress.
And turns it into power.
It generates megawatts of electrical power for the submarine systems simply by existing under the weight of the ocean.
Or the text mentions aerospace, right?
Yes, hypersonic aircraft.
A hypersonic aircraft flying at Mach 5 generates brutal aerodynamic friction and vibration that normally threatens to tear the ship apart.
It's a massive engineering hurdle.
But if the skin of the aircraft is an engineered amyloid lattice, it absorbs the vibrational stress, prevents structural fatigue, and converts the violent friction of the atmosphere into electricity to run the radar systems.
The stress that normally destroys the machine becomes the fuel that powers it.
I want to paint a picture for you listening of what this looks like in daily life. Imagine your electric car 10 years from now.
It'll look very different.
It doesn't have a 1,000-pound lithium-ion battery bolted to the bottom. In fact, it doesn't have a battery pack at all. The structural frame of the car, the chassis itself, is an algae-grown, uranium-laced bone structure.
Generating power continuously.
As you drive, it generates baseline power from its own internal atomic decay. And as your tires hit potholes and the frame vibrates, the piezoelectric structure turns the mechanical vibration of the bumpy road into acceleration power.
It's a closed-loop system.
Your car's structural skeleton is its battery, and it was grown in a vat using sunlight.
And if you get into a fender bender, the impact shatters the rigid fibers in the bumper, which instantly exposes billions of fresh templating seeds.
Oh, right, the self-repair.
With a small spray of monomer liquid, the bumper slowly regrows and repairs its own physical geometry over the next week.
It is absolute magic masquerading as biology.
It really is.
But it leads to the ultimate question of scale. We have a structural, computing, power-generating material that we can grow in a bioreactor from carbon dioxide, water, and sunlight.
The ultimate building block.
If the manufacturing bottleneck is completely removed, what stops us from scaling this into space?
The short answer is, the laws of physics do not stop us.
Wow.
And that realization forces us to transition from terrestrial engineering to stellar macro engineering. We arrive at the Dyson swarm and the concept of the Type 3 biological node.
Okay, for those who aren't familiar, a Dyson swarm is a theoretical megastructure where a civilization launches millions of solar collectors into orbit around their sun, essentially creating a massive spherical net to capture 100% of the star's energy output.
Right. It's the ultimate energy harvesting project.
But physicists usually point out a fatal flaw in actually building one, don't they?
A materials bottleneck.
Right?
Space is big and stars are massive. To build enough solar panels to encapsulate a star, you require a surface area that is astronomically large.
Like incomprehensibly large.
If humanity dismantled Mercury, Venus, Mars, and every single asteroid in the solar system, and refined 100% of that mass into steel and silicon, we still wouldn't have enough material.
We physically lack the atoms.
And the energy required to mine, smelt, forge, and launch a Dyson swarm far exceeds the total energy available to the civilization before the swarm is built. It is an economic and physical paradox.
You can't bootstrap a forge that big. But the amyloid framework completely circumvents the forge, doesn't it? We don't need to melt down planets.
We replace the industrial supply chain with biological exponential replication.
How does that work in space?
The framework proposes that instead of launching trillions of tons of refined metal, an advanced civilization simply launches a single, compact seed package the size of a satellite.
A satellite, okay.
They target a carbonaceous asteroid in the habitable zone of a star system.
And what exactly is inside this seed package?
A starter culture of highly engineered cyanobacteria.
Just algae.
Photosynthetic algae that have been genetically programmed with a complete blueprint for the entire amyloid technology stack.
So all 8 layers of the computer, the battery, everything.
Yes. The probe lands on the asteroid. The bacteria deploy into the ice and carbon dirt. They use the starlight to photosynthesize, breaking down the asteroid's raw elements.
And they begin excreting the monomers that self-assemble into massive piezoelectric metallized amyloid collector panels.
It eats the asteroid.
It eats the asteroid. And because it's a biological system, it follows the rule of exponential growth.
Exactly. One solar panel replicates its seeds to grow 2. 2 become 4. 4 become 8.
It's paying its own construction costs using the starlight it's harvesting. It just sweeps through the asteroid belt, converting dead rock into an organized, conductive swarm.
The swarm grows itself autonomously.
But the framework takes a massive leap past simple energy collection here. The swarm isn't just a passive array of mirrors, is it?
No, it's highly active. Because the amyloid fibers are metallized and maintain their native helical twist, they act as massive chiral electromagnetic waveguides.
Which means they can direct energy.
When networked together by their internal optical computers, the trillions of collector panels form a coordinated, solar system-sized electromagnetic phased array.
A phased array that big, what can it do?
They can project coherent, shaped magnetic fields back inward toward the star itself.
Hold on. You're saying the swarm can physically manipulate the star?
Actively manage it, yes.
Uh-huh.
By projecting immense inward-facing magnetic pressure, the swarm acts like a stellar-scale tokamak. It can actually compress the stellar plasma.
That's terrifying and amazing.
Furthermore, the framework introduces helioseismology.
Acoustic waves. Sound in a star.
Yes, Stars naturally ring like bells. Acoustic pressure waves ripple through their plasma cores constantly.
Okay.
Because the amyloid swarm is highly piezoelectric, it can physically pulse its massive panels, projecting coordinated resonant acoustic waves deep into the stellar core.
So they are squeezing the star using giant magnetic fields and sound waves to do what?
To throttle the fusion rate of the
It is
star.
to throttle a sun.
If the civilization needs an energy spike, they use magnetic compression to squeeze the core, increasing the density and ramping up the fusion output.
And if they don't need the energy?
If they want to conserve fuel, they project acoustic waves to suppress the natural convection currents inside the star. This starves the core of fresh hydrogen, slowing the burn rate down.
They can control the lifespan of a star.
They can extend the lifespan of a star by tens of billions of years. The star is no longer a wild natural fire. It is an actively managed internal combustion engine.
That alone is mind-bending. But the text included another physics concept that took this from managing a star to managing a galaxy. The Gertzenstein effect. I had to look this up because I thought it was purely science fiction.
It is pure relativistic physics.
Okay, explain it.
In 1962, the Soviet physicist M.E. Gertzenstein published a paper proving that in the presence of a strong, static magnetic field, an electromagnetic wave like light or radio can be converted directly into a gravitational wave.
Turning light into gravity.
Exactly. At human laboratory scales, the conversion efficiency is virtually zero. The magnetic fields required are just too large.
But at the scale of a Dyson swarm.
A Dyson swarm, operating as a coordinated phased array, is generating a rotating magnetic field at the scale of the solar system. At that scale, the swarm acts as a macroscopic Gertzenstein transducer.
So the swarm takes the energy of the star, turns it into a massive magnetic field, and then pumps lasers through it to generate ripples in the fabric of space-time. Gravitational waves? Why?
For zero-loss interstellar communication.
How is it better than radio?
If you try to send a radio signal or a laser across the galaxy, it gets scattered by dust clouds, absorbed by nebulas, and distorted by plasma.
Right, space is messy.
But gravitational waves do not interact with electromagnetic matter. A gravity wave passes straight through a dust cloud, straight through a planet, completely unimpeded.
So it's a perfect signal.
A civilization using Gertzenstein converters at the Dyson swarm scale can establish a flawless, instant, galaxy-wide broadband network.
Which fundamentally rewrites our understanding of cosmic civilizations. I mean, in astrophysics, we use the Kardashev scale.
Right, the energy scale.
Type I controls the energy of a planet. Type 2 controls a star. Type 3 controls an entire galaxy. We have always assumed that becoming a Type 3 civilization requires building millions of giant metal spaceships, conquering planets, and strip mining whole sectors of space over millions of years.
Enfield's framework suggests that assumption is a projection of our own primitive industrial mindset.
Because we only know how to build with forges.
Exactly. The Kardashev scale is no longer about the size of your factories. It's about the density of your biology.
Yeah.
Because the seeded cyanobacteria contains the entire technological stack in its genetic code: the computer, the power plant, the lithography, the swarm architecture. The infrastructure is no longer separate from the organism.
The blueprint is the factory.
Yes. The moment that single seed package survives on a foreign asteroid and begins replication, it is instantly a Type 3 galactic node.
It's instantaneous.
The only bottleneck between a single bacterial cell and a star-throttling Dyson swarm is simply time and local carbon. The geometry literally writes itself into the universe.
But listen, as awe-inspiring as a self-writing universe sounds, we have to pause here.
Okay, what's up?
Because the sheer scale of autonomous power, this exponential growth and self-replication, it sounds absolutely terrifying to a normal human being.
It is a lot to process.
We are talking about unkillable, self-assembling biological material programmed to relentlessly convert dead matter into infrastructure. We need to address the glaring civilization-ending elephant in the room. Let's dig into the descent. How do we know this is safe, scalable, and actually feasible outside of a theoretical physics paper?
The skepticism is absolutely necessary. And the most prominent technical criticism is the integration fantasy objection.
The integration fantasy.
Critics will look at the framework and say, yes, DNA computing works in a beaker. Yes, terahertz unzipping works in a simulation. Yes, amyloid is piezoelectric. But you have never integrated all 8 layers into a single working organism.
And if you stack that many complex things...
If you stack 8 bleeding-edge, highly sensitive experimental technologies on top of each other, the complexity overhead guarantees catastrophic failure.
It's the classic, it works in the sterile lab, but the real world will break it immediately argument.
Right. But the rebuttal from the framework's engineers relies on the fact that this is not a fragile serial chain like a silicon microchip.
Because if one transistor fails on a silicon chip,
The entire sector of the processor faults. It crashes. But the biological computer is a parallel, redundant architecture.
meaning if one layer breaks, the others catch it.
It degrades gracefully. Let's go back to your excellent pushback about the terahertz clock signal on the water boiling.
Okay, yeah.
Let's say the environment gets completely chaotic, the thermal ramps fail, and the terahertz unzipping breaks down entirely. The system doesn't crash.
It doesn't blue screen,
The terahertz layer simply goes offline, and the computer defaults to Brownian computing.
which is what, again?
It uses the ambient, random thermal fluctuations of the water to slowly nudge the logic gates forward. It slows down from picosecond processing to minute-long processing, but it remains thermodynamically favored to find the correct logical answer eventually.
So it slows down, it doesn't die.
Exactly.
Okay, what about the industrial scalability? Critics say, sure, a Cambridge PhD student can grow a 10 nanometer aligned fiber in a perfectly still test tube, but you are proposing growing millions of tons of this stuff to replace steel beams.
The scaling problem, yes.
If you try to mix this in a 10,000-gallon industrial vat, the fluid dynamics and chaotic mixing will just create a useless, tangled ball of sludge. You can't align miles of microscopic fiber in a giant blender.
The answer to the batch mixing limit is quite simple. You don't use batch mixing.
Oh, what do you use?
Chemical engineers solved this years ago with continuous flow microreactor.
Microreactors.
Instead of a giant vat, you pump the raw monomer liquid through millions of parallel microscopic tubes. As the fiber grows and flows down the tube, you apply an inline magnetic field.
And that straightens them.
Because the fibers have a natural diamagnetic anisotropy, meaning they naturally align themselves with magnetic field lines, they perfectly straighten out as they gel into a solid.
So the scale transition is entirely manageable.
It is an established industrial process just applied to a new polymer.
But here is the massive one, the containment and evolution objection.
Yes, the mutation fear.
If we build our entire civilization's infrastructure, our homes, our cars, our data centers, using a self-replicating prion bioreactor, what stops it from mutating?
That's a fair question.
What stops the structural beam in my living room from experiencing a random mutation and evolving into an airborne human lethal brain pathogen?
This requires a thorough dismantling because the fear stems from conflating living Darwinian organisms with non-living thermodynamic polymers.
But they replicate.
Fire replicates, crystals replicate. That does not mean they have a genome. Amyloid fibers, the actual prions forming the beams and computers, are not alive. The prion fiber does not contain DNA. It cannot transcribe new information. Therefore, the fiber itself cannot mutate on its own.
So it's locked.
It is merely a thermodynamic shape. It grows by recruiting monomers that exactly fit its geometric lock and key template. It is chemically blind to anything else.
Okay, so the structural beam can't mutate. But what about the cyanobacteria? The biological algae in the vats that are reading the DNA and manufacturing the raw protein monomers, those are alive. Those can mutate.
They absolutely can, and that is why the biological manufacturing layer is completely physically isolated from the structural layer. We already know exactly how to handle mutating bacteria globally.
You mean biosafety levels?
Precisely. Today, right now, the vast majority of human insulin used by diabetics globally is manufactured in massive industrial bioreactors filled with genetically modified E. coli bacteria.
No, I didn't know that.
We know how to secure a bioreactor. We apply standard biosafety level containment to the cyanobacteria host cells.
So they never leave the factory.
The host cells stay in the secure vat. They excrete the inert protein monomers. Only the non-living, non-mutating amyloid fiber comes out of the reactor to be used in your living room. The risk of pathological evolution is entirely managed through standard existing biosafety protocols on the host organism.
Okay, that clarifies the biological safety. But I have one more pushback, and this one is a bit more philosophical.
Let's hear it.
Earlier, we used the Lego brick analogy, and you mentioned the term physical hologram. The source text makes a huge deal about the cross-beta architecture being a hologram. It literally compares a 10-nanometer protein string to the event horizon of a black hole.
It does make that comparison.
I'm sorry, but is it really fair to make that comparison? Isn't that just a physicist getting overly poetic? It sounds like massive philosophical overreach.
It sounds like hyperbole until you look at the underlying mathematics of information geometry. It is not a metaphor. It is the exact same mathematical principle.
How is a protein like a black hole?
In theoretical physics, the Bekenstein-Hawking formula for black hole entropy resolves a massive paradox about information falling into a black hole.
Right, the idea that information can't be destroyed.
Stephen Hawking and Jacob Bekenstein proved that the total amount of three-dimensional information inside a black hole, everything that ever fell into it, is perfectly mathematically encoded on its two-dimensional surface area, the event horizon.
The 2D surface dictates the 3D volume.
That is the holographic principle. Now look at the cross-beta fibril.
Okay.
The entire architectural blueprint for the three-dimensional length of the fiber, whether it's a nanometer long or a mile long, is perfectly mathematically encoded and dictated by the 2D surface area of the fibril tip.
The tip of the string.
The specific arrangement of hydrogen bonds and amino acids on that microscopic flat surface contains the total instruction set for the entire structure that will grow from it.
The part contains the whole.
The sequence encodes the fold. The fold encodes the 2D surface. The 2D surface strictly dictates the 3D fiber. It operates on identical information geometric logic to the Bekenstein-Hawking formula, just scaled down by 35 orders of magnitude.
The geometry is the information.
It is literally a physical hologram.
That is just, it really forces you to reevaluate the line between biology and physics.
It erases the line entirely.
Having defended the science, I want to bring you, the listener, back down to earth. We have gone on an incredible stretching journey today.
We really covered the spectrum.
We started by looking at a 10-nanometer protein fiber, something humanity has historically only viewed as a terrifying biological error. We watched it become an indestructible, laser-printed architectural beam.
And then a computer.
Right. We saw it transform into a heat-powered 8-layer quantum computer. We watched it be woven with uranium to create waste-free nuclear batteries. And we conceptually scaled it up to a Dyson swarm using acoustic waves to manipulate the fusion of a star.
It represents a complete rewrite of how humanity interacts with the physical universe.
It really does. I want you to look around your physical environment one more time. Look at the concrete walls, the steel girders out the window, the silicon processing power in the screen you are staring at.
All extracted materials.
Every single piece of that infrastructure required violence against the earth to create. This framework, this staggering vision synthesized by Enfield, tells us that the future of infrastructure won't be forged in fire. It will be seeded in water, illuminated by precision light, and allowed to grow.
The rigid boundaries we draw between material, machine, and organism will dissolve entirely into a single programmable geometric reality.
And I want to leave you with one final, deeply provocative thought to mull over. We talked extensively about the galactic seed package.
The Type 3 node concept.
Yeah. The idea that a truly advanced Type 3 civilization wouldn't waste time building giant metal starships to conquer the galaxy. They would simply compress their entire computing, architectural, and energy harvesting infrastructure into a microscopic, self-assembling biological seed.
And scatter them across the cosmos.
Letting them crash onto asteroids and planets, waiting for them to replicate using the local chemical soup. It is the most thermodynamically efficient deployment strategy imaginable for a galactic civilization.
Without a doubt.
Now think about our own planet. Think about abiogenesis, the mysterious moment dead chemistry suddenly became life. Or think about the Cambrian explosion, where complex, architecturally diverse biology suddenly and violently appeared in Earth's oceans in a geologic blink of an eye.
Where are you going with this?
If an advanced civilization can compress its entire technological infrastructure into a self-replicating biological seed, how do we know with absolute certainty that Earth's own biology wasn't simply the arrival of a Type 3 civilization's seed package?
Wow.
Are the intricate proteins functioning inside our bodies right now, the very building blocks of our cells, actually the forgotten, dormant infrastructure of an ancient galactic network?
It entirely reframes our search for extraterrestrial intelligence. We may not need to look up at the stars with radio telescopes.
Right.
We may just need to look closer at the geometry inside our own cells.
It really makes you wonder if we are the builders or if we are just the building material waking up. Thank you so much for joining us on this deep dive. Never stop questioning the limits of biology and the limits of technology. We'll catch you next time.
Okay, so what if the deadliest protein known to science was also secretly the key to building our entire future? Today, we're diving into a technology that could genuinely change everything: prion architectures.
This one question, this one paradox, is really what this whole explainer is about. It's an idea that could completely redefine how we build our world, from the tiniest little microchips all the way up to entire cities that can literally repair themselves.
So, to really get this, we have to start with the villain of the story, right? We have to talk about the prion. Yeah, when you hear the word prion, you probably think of something pretty terrifying, and for good reason. We usually only hear about them as these unstoppable agents that cause these devastating brain diseases by triggering a chain reaction of misfolding that, well, can't be stopped.
But here's where it gets really interesting. Scientists are starting to flip the script. They're asking a totally different question, one that reframes the whole problem. What if this unstoppable chain reaction isn't a destructive curse, but an incredible building principle that we've just misunderstood?
So the big reveal here is that the secret isn't in the specific disease-causing protein itself. Not at all. It's in the remarkably powerful structure that it forms. And this structure is like a universal blueprint that makes everything else possible.
This right here is the true hero of our story, the cross-beta architecture. Just think of it like a kind of molecular Lego. It's a universal design that can get almost any protein to snap together and form these ultra-strong, perfectly ordered fibers. It's not a single material. It's a design logic for making materials.
And check this out. This chart just shows you how powerful that structure really is. These fibers are called amyloids when they form this way. We're looking at their toughness, basically how much energy they can absorb before breaking. Now you'll see, they don't just match spider silk, which is kind of the gold standard, they actually beat it. And they do that while being way, way less dense than steel.
And this is the part that sounds like magic, but it's real science. How do you make it? Well, you don't need some huge high-tech factory. You just drop a tiny little seed, a fragment of the fiber, into a solution of proteins. That seed acts as a template. The fibers grow, they break apart, and then each new piece becomes a brand new seed. It's a cascade reaction. The material literally manufactures itself, exponentially. So let's just recap the three big superpowers we're dealing with here. First, you've got incredible strength, and it's lightweight. Second, it's nearly indestructible. It can survive crazy heat, radiation, you name it. And third, and this is the big one, it can build itself. That combination makes it unlike anything else we've ever known.
All right, so we've got this incredible material. What can we, do with it? Well, this is where it starts to sound like straight-up science fiction. But all of this is grounded in research that's happening as we speak.
Think about this for a second. An airplane wing that repairs its own tiny fractures. If a crack starts to form, the broken fiber ends are suddenly exposed to the raw protein sitting in the matrix around them, and they just start regrowing. They literally heal the damage all on their own.
And it's not just big stuff. We're talking about revolutionizing electronics. Instead of carving circuits out of silicon in these massive, complex facilities, you could have conductive amyloid fibers literally grow themselves into the exact patterns you need. The wire just assembles itself from the bottom up.
This goes way beyond just, your typical plant-based burger. This is about building food from a molecular blueprint. By controlling how these fibers self-assemble, you can construct the precise lattice that gives a steak its unique chew, or a piece of shrimp its signature snap.
Okay, but wait a minute. This all leads to a huge question, right? If this stuff grows and replicates exponentially, how in the world do you control it? How do you tell it where to grow and maybe even more importantly, where to stop?
So on their own, these two technologies have pretty big limitations. On one hand, you've got this amazing prion self-assembly that can build massive structures, but you have no control. It just grows everywhere. On the other hand, you've got optogenetics, the science of controlling proteins with light, which gives you incredible precision, but you can't really build anything big with it.
But what if you smash them together? And that's exactly what scientists figured out how to do. They've designed this new protein that fuses the prion-forming part to a light-sensitive switch. So in the dark, it's totally inert, nothing happens. But the moment you shine a beam of light on it, that switch flips, and the self-assembly cascade kicks off, but only exactly where that light is shining.
So what you get is, well, it's literally a light switch for manufacturing. You can use a laser or a complex holographic light pattern to tell these fibers exactly where to grow with microscopic precision. You're basically turning a bulk chemical process into a programmable three-dimensional printer that uses light as its ink.
Okay, so let's zoom out. What's the end game here? We're not just talking about making better gadgets or materials. We are talking about a technology on a civilizational scale, a completely new foundation for how we build our world.
This is the big idea, right? We're talking about a fundamental shift from a built world to a grown world. An infrastructure that can be seeded, that can repair itself, that's manufactured from renewable resources using light as the main tool. It's this single, unified platform that could connect everything from our food to our computers.
And that really leaves us with this one kind of profound thought from the source material. We might be on the edge of discovering one of the most powerful building blocks in the universe, a technology that blurs the line between a material and a machine, between an organism and infrastructure. The real challenge is going to be understanding what we're holding.